Yeah, maybe we can try to clean it up today. Okay. Oh, he go heal. Probably. Jesus, I got stuck to it. Oh, my bad. What the heck? What is it saying? I can't read it. It's so small. Oh, that's a bit too much, eh? We're at 150? Yeah, 150. Was it 150? Oh, no. Here's 175. What the heck? Oh, yeah, hello. Oh, that's right. It doesn't matter. It does. Excuse me. Oh, major. It's ground control to Major Tom. Ground control to Major Tom. Okay, wait. What's it gonna say? Uh, to switch regions. Oh. Oh, actually, that's good. Excuse me. Ah. Okay. I think maybe that's better. Oh. Fusion forces. Fusion forces and a change in density. Oh, wait, wait, wait. Why don't we just kind of relax a little bit? We'll take a 5 minute before we begin the videos. At least we made it this far, so I'm happy. Hey Akash. Hey Zen. Yeah, Cat Academy. This Cat Academy course is free. Just type in Cat Academy. Go over to where's it at? Go over to MCAT right here. Yeah, go over to MCAT and you should be okay. You know? But yeah, it's all free. Free. Go back. Oh, seriously? Why isn't I going back? Oh, there you go. What is the MCAT? MCAT is medical college admission test. It's a test required to get into medical schools to become a doctor. A doctor. With that said, I'll be back. Let me just start. right, I'll be back. Um, I'm all right. It's okay. It's okay. It's only a game. Why you have to be so mad?
Point of the immune system is to keep out shady things from your body, or if they get in, to kill them. So that those shady things would include shady proteins that can do damage to your body, viruses, bacteria, even eukaryotic parasites, and then even fungi. So all sorts of things that if they were to enter your body, they would cause some form of disease. These are collectively called pathogens. So the whole point of the immune system is, on a first line of defense, keep these things out, and then if they were to get into your body, to kill and eliminate them from our system so that we don't get sick and so that we don't die. So I already just mentioned that there's kind of two lines of defense, and even with system, immune system is to keep out shady things from your body, or if they get in, to kill them. So that those shady things would include shady proteins that can do damage to your body, viruses, bacteria, even eukaryotic parasites, and then even fungi. So all sorts of things that if they were to enter your body, they would cause some form of disease. These are collectively called pathogens. So the whole point of the immune system is, on a first line of defense, keep these things out, and then if they were to get into your body, to kill and eliminate them from our system so that we don't get sick and so that we don't die. So I already just mentioned that there's kind of two lines of defense, and even with those, there's kind of sub-classifications. The first line of defense, I'll just call that the first line, which is essentially just to keep things out. Keep all of these pathogens out. And there's some obvious ones. There's our skin. Our skin keeps pathogens out, and actually even the oils on our skin are a little bit more acidic, and it's hard for some types of bacteria to thrive in that type of an environment. You have your mucous membranes, and in the mucus there's some chemicals that maybe make it a little bit more difficult for bacteria to survive. And then you even have acidic environments like your stomach acid. Keep that in the same color. Stomach acid. You might not view your stomach as the outside of you, but it fundamentally is. Your whole digestive tract, which I'll make videos on in the near future, is really on the outside of you. You can simply model really most vertebrate bodies as kind of a donut, and our digestive tract is kind of the inside of the donut. So stomach acid is on the outside of our real bodies, and you can imagine that's a hard environment for a lot of these pathogens to survive in. So that's the first line of defense, but we know that that's not good enough, that sometimes these things can get into our bodies, and there we have to start thinking about the second line of defense. What do we do once things are actually in our body? Second, let me switch colors. So then you have your second line of defense. Second line. And here, in both the first and second line, I'm talking about non-specific immunity. And this is going to make a lot of sense when we start talking about specific immunity. So both of these are non-specific. And when I say non-specific, or you can also call them innate, it means that they just generally respond to things that appear bad. They don't remember the bad things that came before. They don't respond to a particular type of virus or they do respond to every type of virus or every type of bacteria, but they don't say, oh, this is virus type, ABC, or this is bacteria type ABC. They just say, this is a virus, let me get rid of it, or let me not let it in. This is a bacteria, let me get rid of it, or let me not let it in. It doesn't know what type of bacteria it's dealing with. So this is all the non-specific or innate immune system. And we'll go into a lot of detail onto the specific immune system, because you can imagine it becomes very complicated or interesting when you start thinking about your body somehow remembering a virus that it's seen before and being able to respond better to that virus or that bacteria or that protein the second time it sees it. So we're dealing with non-specific in this case. And the second line of your non-specific immunity, there are two things. One is an inflammatory response. Inflammatory response. And I'm going to do a whole video on this, but in general, we've all experienced inflammatory responses when you see blood flowing to a certain part of an area and you see there's pus and I'm going to go into a lot more detail on what an inflammatory response actually is, but that's one of and what it really is doing is bringing blood and bringing cells that can fight whatever type of infection you have. It's bringing them to the site where maybe you got a cut, or maybe where a lot of the bacteria or whatever the pathogen is. So inflammatory response is all about bringing fluid and fighters to the fight. So bringing stuff to the fight. Let me write that. And I'm going to do a whole video on that. Bringing stuff to the fight. You can almost imagine bringing weapons to the fight. But the byproduct is that part of your tissue or that part of your body gets inflamed. A lot of fluid there, a lot of, I guess, byproducts of the battle that goes on there. We'll do a whole video on that. And then the other second line of defense, and it's actually part of the inflammatory response, are phagocytosis, or phagocytes. And really what I want to do over the rest of this video is talk in a little bit more detail about phagocytes. Because once we understand what phagocytes do, that's a pretty good building block for going into the specific immune system. And actually, it'll help lead into the discussion on the inflammatory response as well, because phagocytes are really part of the inflammatory response. So phagocytes are just a class of cell that can eat up pathogens. They can eat up other things, really, but when we talk about the immune system, we're talking about pathogens. So let's say that this is a phagocyte right here. This is a phagocyte right there. It has some kind of a nucleus, whatever. I don't have to focus on the inside of the phagocyte. It's a traditional eukaryotic cell. But what I want to do is see what happens when a phagocyte encounters a foreign particle or a foreign bacteria. So let me say this is a foreign bacteria right here. So the phagocyte, we've already said, is non-specific. What it does is it has receptors that respond to just things that it knows are bad. You can imagine these are super sensors, so maybe these are super sensors right here. These are super sensors for bacteria. And the bacteria have proteins on their surface that maybe look something like that. Obviously, they don't look exactly like that. I'm just drawing them as kind of a Y and a triangle so you can see that they fit. But once these two guys connect, so let me draw the situation where they have connected. So this is the bacteria, this is the pathogen, and it's really the same idea with a virus or any other type of thing. And we'll actually see in future videos that these guys can actually be tagged by other molecules, which makes these phagocytes want to attack them even more. Well, let's say once they're bonded, so that's the bacteria, the invading pathogen, and now it is bonded. It has triggered the receptor on this phagocyte. This phagocyte will start to engulf. It'll wrap around this pathogen. So it'll start wrapping around the pathogen like that. And these two ends are going to eventually going to meet. They're eventually going to meet, and I can still let me draw the nucleus in a different color so we know it's not really related to the phagocytosis. So maybe that's the nucleus, and there's other organelles sitting in here. But then once these two meet, what's it going to look like? Then all of a sudden, that bacteria is going to be completely engulfed. It's going to be inside of the cell. So now the cell, once these two ends meet and these membranes merge, then this guy is going to be in his own little membrane bubble, or you can almost imagine it's in its own little vesicle. So this is the pathogen, the bacteria in this case, but the phagocytosis, the process is completely identical in terms of how it engulfs things. If it was a virus or some type of other foreign protein or any type of really foreign molecule, and And actually, sometimes it doesn't even occur to foreign stuff. It can occur to stuff that is dying molecules that are not foreign, that just need to be cleared out. But we'll just focus on the immune system, on foreign things right now. So it was merged. Let me draw. So then this membrane right here will completely merge and go around this guy like this. And of course, you had your receptors, and who knows if they're still there by the time. Let's just draw them there so you see that that part is that part. But once it's fully engulfed, this thing is called a phagosome. This is a phagosome. Phagosome, which is really just a vesicle that contains that foreign particle that you want to get rid of. And then other fluid or vesicles that contain, let me draw it in a different color, that contain things that can eat up this phagosome. So let's say that, here, right, this is some vesicle that contains things, lysozymes, and it contains really reactive species of oxygen. So let's say that, it contains a bunch of stuff here. And if this comes in contact with really almost any biological compound, it's going to do some damage. But once the pathogen is completely merged inside the cell, this little package will merge over here, and it will dump its contents into this phagosome, into this vesicle containing the pathogen, and then break it up. It's essentially digesting it. So obviously the first role is it just got it out of the way and it killed it. And then the second role, and I'm just going to give a little tidbit right here, and we're going to do it in a lot more detail in future videos. It breaks it up. So let me draw the whole thing now. I want to keep my colors consistent. It'll break it up. So now the thing is all broken up. It's all broken up. This is the membrane inside. So that thing is broken up into constituent proteins and other molecules. And then what the phagocyte does, it'll actually take some subset of these molecules, some subset of the proteins. It'll break them down. Proteins are just sequences of amino acids. So normally when people say proteins, they're talking about long sequences of amino acids. When people talk about short sequences of amino acids or a protein that's broken up a lot, they refer to it as a peptide chain. A peptide chain is a shorter chain of amino acids. So this guy will take some special peptide chains, some special pieces from the thing it just killed, attach them to some other proteins. So it'll take maybe a little piece of this bacteria right now, attach it to other protein. Let me just do that in pink. It'll attach it to this other protein, which is called a major histocompatibility complex. And if we're talking about phagocytes, this will be a major histocompatibility complex type 2. And that's a mouthful of words. So this is it sounds very a strange word, but we're going to see this a lot. So this is major histocompatibility complex. So they abbreviated MHC. This is a protein, and it bonds with this peptide that was kind of chunked off or digested off of this invading pathogen. And then this phagocyte will then present it onto its membrane. So this combination, the complex of the MHC, and in this case it's going to be an MHC2 protein. We're going to talk, so MHC type 2. We're going to talk about type 1 in the future. It's going to take this complex and then present it on its surface. And then it's going to present the combination of the MHC type 2 and the little peptide chain that came from the thing, and present it on its surface. And the reason why I'm going through all of this pain of explaining this process, you're like, hey, we already got rid of the thing and killed it. Why is Sal worried about what we do with the peptides? This is crucial to our immune system because we'll see other specific parts of our immune system. Remember, so far everything is non-specific. This guy just said, oh, this is an invader. It doesn't know the type of an invader. It just says, hey, let me bond to this thing and kill it. It's not. It's one of these things that I know are foreign to my body. So it kills it, but now it can leave it on its surface, and now the specific parts, the parts that actually have memory and attack specific things, can say, gee, Mr. Phagocyte, look, you've killed something. Let me see if I have some specific reactions that can be triggered by this thing that you're presenting. So many phagocytes are also called antigen presenting cells. Antigen presenting cells. And I'm going to go into more detail on what exactly an antigen is. I called this thing a pathogen. An antigen is essentially, you can view it as a protein or a peptide chain that will trigger or that can be dealt with within the immune system. I'll be a little bit more the specific immune system, and I'm going to be a little bit more nuanced about it when I talk. I'll make a whole video on antigens and antibodies, but right now you can just view it as a peptide chain right there. An antigen is just a protein or a part of a protein. So this is presenting an antigen on its surface that can later be used by other parts. Now, the one thing that there are many types of phagocytes, and just to give you just so when you see different words you don't get confused by the different types of phagocytes, I'll do a little review of those right now. You have neutrophils. These are actually the most common of the phagocytes, and these are kind of the fast and numerous responders. So these get to a location of infection very fast. So fast and abundant. And phagocytes don't necessarily just have to kill in this way. I mean, they're called phagocytes because they engulf this way, but we'll in future videos talk about other ways that they can release chemicals or even DNA nets to ensnare pathogens. But neutrophils are fast and abundant. And then you have macrophages, which are kind of on some level, they're the most versatile and do the heavy lifting, but they're also phagocytes. And then you have dendritic cells. And when you first see the word dendritic cell, you think, hey, does this somehow relate to dendrites of the nervous system? And no, they have nothing to do with the nervous system. Nothing nervous system about them. Nothing nervous. The reason why they're called dendritic cells is because they look like they have dendrites. They'll look like, they have these little things coming off of them, just like that. And if you kind of just view it as a division, so they look like neurons on some level, but they don't participate in the nervous system at all. And these tend to be the best activators of the specific immune system that we'll talk about in future videos. So anyway, I'll leave you there, and we'll talk more about all of this in the next few videos. In the last video, we talked a little bit about the. Let's just talk. In the very first, in talking about.
Jeez. Okay. Actually, let's go back a little bit then.
Fat draw. What's it going to look like? Then all of a sudden, that bacteria is going to be completely engulfed. It's going to be inside of the cell. So now the cell, once these two ends meet and these membranes merge, then this guy is going to be in his own little membrane bubble, or you can almost imagine it's in its own little vesicle. So this is the pathogen, the bacteria in this case, but the phagocytosis, the process is completely identical in terms of how it engulfs things. If it was a virus or some type of other foreign protein or any type of really foreign molecule, and actually sometimes it doesn't even occur to foreign stuff. It can occur to stuff that is dying molecules that are not foreign, that just need to be cleared out. But we'll just focus on the immune system, on foreign things right now. So it was merged Let me draw. So then this membrane right here will completely merge and go around this guy like this. And of course, you had your receptors, and who knows if they're still there. But a fluid or vesicles that contain. Let me draw it in a different color. That contain things that can eat up this phagosome. So let's say that here, right, this is some vesicle that contains things, lysozymes, and it contains really reactive species of oxygen. So let's say that it contains a bunch of stuff here, and if this comes in contact with really almost any biological compound, it's going to do some damage. But once the pathogen is completely merged inside the cell, this little package will merge over here, and it will dump its contents into this phagosome, into this vesicle containing the pathogen, and then break it up. It's essentially digesting it. So obviously the first role is it just got it out of the way and it killed it. And then the second role, and I'm just going to give a little tidbit right here. We're going to do it in a lot more detail in future videos. It breaks it up. So let me draw the whole thing now. I want to keep my colors consistent. It'll break it up. So now the thing is all broken up. It's all broken up. This is the membrane inside. So that thing is broken up into constituent proteins and other molecules. And then what the phagocyte does, it'll actually take some subset of these molecules, some subset of the proteins. It'll break them out. Proteins are just sequences of amino acids. So normally when people say proteins, they're talking about long sequences of amino acids. When people talk about short sequences of amino acids or a protein that's broken up a lot, they refer to it as a peptide chain. Peptide chain is a shorter chain of amino acids. So this guy will take some special peptide chains, some special pieces from the thing it just killed, attach them to some other proteins. So it'll take maybe a little piece of this bacteria right now, attach it to other protein. Let me just do that in pink. It'll attach it to this other protein, which is called a major histocompatibility complex. And if we're talking about phagocytes, this will be a major histocompatibility complex type 2. And that's a mouthful of words. So this is it sounds very a strange word, but we're going to see this a lot. So this is major histocompatibility complex. So they abbreviated MHC. This is a protein, and it bonds with this peptide that was kind of chunked off or digested off of this invading pathogen. And then this phagocyte will then present it onto its membrane. So this combination, the complex of the MHC, and in this case it's going to be an MHC2 protein. We're going to talk, so MHC type 2. We're going to talk about type 1 in the future. It's going to take this complex and then present it on its surface. And then it's going to present the combination of the MHC type 2 and the little peptide chain that came from the thing and present it on its surface. And the reason why I'm going through all of this a pain of explaining this process, you're like, hey, we already got rid of the thing and killed it. Why is Sal worried about what we do with the peptides? This is crucial to our immune system because we'll see other specific parts of our immune system. Remember, so far everything is non-specific. This guy just said, oh, this is an invader. It doesn't know that. Present it onto its membrane. So this combination, the complex of the MHC, and in this case it's going to be an MHC2 protein. We're going to talk, so MHC type 2. We're going to talk about type 1 in the future. It's going to take this complex and then present it on its surface. And then it's going to present the combination of the MHC type 2 and the little peptide chain that came from the thing and present it on its surface. And the reason why I'm going through all of this a pain of explaining this process, you're like, hey, we already got rid of the thing and killed it. Why is Sal worried about what we do with the peptides? This is crucial to our immune system because we'll see other specific parts of our immune system. Remember, so far everything is non-specific. This guy just said, oh, this is an invader. It doesn't know the type of an invader. It just says, hey, let me bond to this thing and kill it. It's not. It's one of these things that I know are foreign to my body. So it kills it, but now it can leave it on its surface, and now the specific parts, the parts that actually have memory and attack specific things, can say, gee, Mr. Phagocyte, look, you've killed something. Let me see if I have some specific reactions that can be triggered by this thing that you're presenting. So many phagocytes are also called antigen-presenting cells. Antigen-presenting cells. And I'm going to go into more detail on what exactly an antigen is. I call this thing a pathogen. An antigen is essentially, you can view it as a protein or a peptide chain that will trigger or that can be dealt with within the immune system. I'll be a little bit more the specific immune system. And I'm going to be a little bit more nuanced about it when I talk. I'll make a whole video on antigens and antibodies. But right now you can just view it as a peptide chain right there. An antigen is just a protein or a part of a protein. So this is presenting an antigen on its surface that can later be used by other parts. Now, the one thing that, there are many, many types of phagocytes. And just to give you, just so when you see different words, you don't get confused by the different types of phagocytes, I'll do a little review of those right now. You have neutrophils. These are actually the most common of the phagocytes. And these are kind of the fast and numerous responders. So these get to a location of infection very fast. So fast and abundant. And phagocytes don't necessarily just have to kill in this way. I mean, they're called phagocytes because they develop this way, but we'll in future videos talk about other ways that they can release chemicals or even DNA nets to ensnare pathogens. But neutrophils are fast and abundant. And then you have macrophages, which are kind of on some level, they're the most versatile and do the heavy lifting, but they're also phagocytes. And then you have dendritic cells. And when you first see the word dendritic cell, you think, hey, does this somehow relate to dendrites of the nervous system? And no, they have nothing to do with the nervous system. Nothing nervous system about them. Nothing nervous. The reason why they're called dendritic cells is because they look like they have dendrites. They'll look like, they have these little things coming off of them, just like that. And if you kind of just view it as a division, so they look like neurons on some level, but they don't participate in the nervous system at all. And these tend to be the best activators of the specific immune system that we'll talk about in future videos. So anyway, I'll leave you there, and we'll talk more about all of this in the next few videos. The immune system. And in that video, we focused on the non-specific or the innate immune system. So let me write that, non-specific immune system. And even in the non-specific immune system, we subdivided that into kind of the first line, kind of barriers. And those were things like the skin or the stomach acid or the acidity of the oils on the outside of your skin. These are just natural barriers to not allowing things inside of your body. But then once they get in, so you can almost imagine these were the first line of defense. And then you had your second line of defense, but these are still non-specific. And when we say non-specific, it means that they don't necessarily know what type of virus, what type of protein, what type of bacteria. They just know that this thing looks shady. Let me eat it up. Let me kill it. Let me have an inflammatory response. So in there, we said, well, there's an inflammatory response, which I'm actually going to do. I'm going to talk about after we do videos on the specific immune system. You have your inflammatory response, which really just gets things to where the action is at. And then you also have your phagocytes, which are these cells that are engulfing things. Phagocytes. And just so you know, all the phagocytes that we talked about in the last video, these are all instances of white blood cells or leukocytes. So these are all instances, I'll write that in white. These phagocytes right here, these are all, I talk about dendritic cells and macrophages and neutrophils. These were all white blood cells. These weren't all the kinds of white blood cells. We're about to talk about more. And sometimes the other word for white blood cell is also leukocyte. So that is non-specific. It just said, well, one, it just doesn't let you in. But then when you're in, it says, hey, you're just shady. I'm going to eat you up. I have receptors. You have some double-stranded DNA floating around. Only viruses have double-stranded DNA. I'm going to eat you up. I don't know what type of virus you are. I don't know if one, it just doesn't let you in. But then when you're in, it says, hey, you're just shady. I'm going to eat you up. I have receptors. You have some double-stranded DNA floating around. Only viruses have double-stranded DNA. I'm going to eat you up. I don't know what type of virus you are. I don't know if I've seen you before or not. That's why it's non-specific. Now, the really interesting thing about our immune system, and this non-specific, this is kind of this exists across many species and types of organisms. But the specific is kind of it's thought to be a newer adaptation. What I'm going to talk about is the specific immune system that's particular to humans. So you have the specific. You have your specific immune system. That's our other classification. We do it like that. So then you have your specific, or you can imagine it's an adaptive immune system. So, and I think you've probably heard of things like that. Oh, I have a resistance to that bacteria or that virus. So this is adaptive. And it's really all based on having exposure to things. And within the specific immune system, we talked when we talked about the antigen presenting molecules that the phagocytes do, that plays a role in this. We're going to go into more detail, but I don't want to confuse you. But the main actors here are called lymphocytes, not to be confused with leukocytes, because they still are leukocytes. So let me write this down, lymphocytes. So these are the main actors. Lymphocytes. And I want, these are specific. Phagocytes, for the most part, are non-specific. But both of these are white blood cells. Lymphocytes are another type of white blood cell or a leukocyte. Don't want to confuse you with this convoluted diagram, but I just want to make the terminology clear. When someone talks about a white blood cell, they're really just talking about a set of cells that when people first tried to separate the components of blood, you'd have your red blood cells that would kind of settle in the bottom, then you'd have this layer of white frothy stuff in the middle that was really made of white blood cells, and then on the top you had the fluid, the plasma from your blood, the kind of the watery part. So that's where the name came from, but they have different roles, but they interact with each other. Now, lymphocytes can be divided into B lymphocytes B lymphocytes, usually referred to as B cells, and T specific. You have macro immune system, you have your inflammatory response, which really just gets things to where the action is at. And then you also have your phagocytes. Phagocytes, which are these things, these cells that are engulfing things. Phagocytes. And just so you know, all the phagocytes that we talked about in the last video, these are all instances of white blood cells or leukocytes. So these are all instances, I'll write that in white. These phagocytes right here, these are all, I talk about dendritic cells and macrophages and neutrophils. These were all white blood cells. These weren't all the kinds of white blood cells. We're about to talk about more. And sometimes the other word for white blood cell is also leukocyte. Leukocyte. So that is non-specific. It just said, well, one, it just doesn't let you in. But then when you're in, it says, hey, you're just shady. I'm going to eat you up. I have receptors. You have some double-stranded DNA floating around. Only viruses have double-stranded DNA. I'm going to eat you up. I don't know what type of virus. This is killer to humans. So you have the specific. Plays a role in this. We're going to go sites. Lymphocytes, and I want, these are specific. Phagocytes, for the most part, are non-specific, but both of these are all white blood cells. Lymphocytes are another type of white blood cell or a leukocyte. Don't want to confuse you with, this convoluted diagram, but I just want to make the terminology clear. When someone talks about a white blood cell, they're really just talking about a set of cells that when people first tried to separate the components of blood, you'd have your red blood cells that would kind of settle in the bottom, then you'd have this layer of white frothy stuff in the middle that was really made of white blood cells, and then on the top you had the fluid, the plasma from your blood, the kind of the watery part. So that's where the name came from, but they have different roles, but they interact with each other. Now, lymphocytes can be divided into B lymphocytes, usually referred to as B cells, and T lymphocytes. You say, wait a minute, where do those words B and T? T lymphocytes. And the B and T just come from where they're developed. B lymphocytes were first recognized in the bursa of Fabricius. So it's called B. That's actually a part of birds that participate in the immune system. And so the B came from bursa. But B also applies to the human immune system because it's produced in bone marrow. So that might be an easier way to remember. It's produced in bone marrow. It's developed in bone marrow. But historically the B came from the bursa of Fabricius, just in case you want to know. But it's easy to remember the B could also stand for bone marrow because that's where it's produced. T lymphocytes actually do start off in the bone marrow, but they mature and become what they are in the thymus. So that's where the T comes from. T for thymus. Now, in this video, I'm going to focus just on the B lymphocytes, because frankly, if I focused on everything, it would be an hour-long video. But the B lymphocytes, frankly, on some level, I don't want to pick and choose favorites, but for something in my brain, I just really like the B lymphocytes. So the B lymphocytes participate in what's called a humoral response. Humoral, and I'll tell you what humoral means in a second. Humoral doesn't mean it's funny. Humoral response. Humoral response, and you'll see that T lymphocytes participate in what's called the cell-mediated response. We're going to do that in a future video. Cell-mediated response. So they actually do certain classes of T lymphocytes. We'll see that there are helper T cells, and then there are cytotoxic T cells. I know it's all very confusing the first time you see it. That's why I just want to focus on just this part right here. We're going to see in the future that the helper T cells play a role in amplifying and really activating this humoral response. But a simple way to think about the difference between the humoral response and the cell-mediated response is when I get infected, let's say I get infected by a virus, right? Let's say, let's say that's my body cell and that's my body cell. At first, when a virus comes into my system, it's just floating around in the fluids in my system. And when the fluids of our system, that's really what humoral responds to, into the humoral fluids of your body. So you have your viruses, do a different color. These are little viruses floating around. So while they're floating around and they're not sitting inside of cells, when they're not sitting inside of cells, that's where the humoral response can come into play. Same thing if we have little bacteria floating around and they haven't infiltrated cells yet, they're just floating around in the fluid, then the humoral response can be useful for that. Now, if all of a sudden these guys have infiltrated cells, so if the cells are now infected with the virus and the virus is using the mechanisms of the cell to produce more, then all of a sudden we have to be a little bit more sophisticated in how we deal with these cells and how we deal with the viruses, because they're not just going to be floating around anymore. We probably want to just kill this cell, even though it was one of our own, but now it's helping to make viruses, or maybe it's been colonized by bacteria. So in either case, you want to kill this, and that we'll talk more about that in the cell-mediated.
Oh, that looks pretty good.
But let's just talk about the humoral response right now that deals with B lymphocytes. So B lymphocytes, or B cells, we do in blue, B for blue. So let's say that is a B lymphocyte. It's a white blood cell. It's a subset of white blood cells called lymphocytes. It comes from the bone marrow. And that's where the B comes from bursa. For bursa, we don't want to go into detail there. But they have all of these proteins on their surface, actually close to 10,000 of them. It's actually, well, I get very excited about B cells. I'll tell you why in a second. So it has all of these proteins on them that look something like this. I'll just draw a couple of them. These proteins, these are actually protein complexes. You can kind of view them. They actually have 4 separate proteins on them, and we can call these proteins membrane-bound antibodies. These right here are membrane-bound antibodies. And I'll talk a lot more about antibodies. You've probably heard the word. Do you have antibodies for such and such flu or such and such virus? And we're going to talk more about that in the future. But you can really just, antibodies are just proteins. Sometimes they'll, hold on.
Yeah. I don't think we'll do it yet.
They're often referred to as immunoglobulins. Immunoglobulins. Teaching biology really stresses the spelling part of my brain. But these are essentially equivalent words. Antibodies are immunoglobulins, and they're really just proteins. Now, B cells have these on the surface of their membranes. These are membrane bound. And usually when people talk about antibodies, they're talking about free antibodies that are going to just be floating around like that. And I'm going to go into more detail on how those are produced. Now, what's really, 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 really interesting about these membrane-bound antibodies and these B cells in particular is that a B cell has one type of membrane-bound antibody on it. Each B cell, right? So that's one B cell. Let me draw another B cell here. So let's say that this is another B cell right here. It's going to also have antibodies, but those antibodies are going to be different. And we'll focus on where they're different. So let me just draw them the same color first, and then we'll focus on where they're different. So that's one antibody, membrane bound. That's another, be different. That's one B cell. And let me draw another B cell here. So let's say really, 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 really interesting about these membrane bound antibodies and these B cells in particular is that a B cell has one type of membrane bound antibody on it. Each B cell, right? So that's one B cell. And let me draw another B cell here. So let's say that this is another B cell right here. It's going to also have antibodies, but those antibodies are going to be different. And we'll focus on where they're different. So let me just draw them the same color first, and then we'll focus on where they're different. So that's one antibody, membrane bound. That's another antibody, membrane bound. These are both B cells. They both have these antibodies on them. Now, the interesting thing is that from one B cell to another B cell, they have a variable part on this antibody that can take a bunch of different that can take on a bunch of different forms. So this one might look like that and that. And these are these long, I'll go into more detail on that, and like that, and that. So the colors, let me do it. So there's the fixed portion, you can find it as green for any kind of antibody, and then there's a variable portion. So maybe this guy's variable portion is, I'll do it in pink. And every one of the antibodies bound to his membrane are going to have that same variable portion. Now, but this different B cell is going to have different variable portions. So I'll do that in a different color. I'll do it in magenta. So his variable portions are going to be different, just like that. Now, he has 10,000 of these on a surface, and every one of these have the same variable portions, but they're all different from the variable portions on this B cell. And there's actually 10 billion different combinations of variable portions. So there's 10 to the 10th, or 10 billion combinations of variable portions. So the first question, and I haven't even told you what the variable portions are good for, is how do that many different combinations arise? Obviously, these proteins, maybe not so obviously, all the proteins that are part of most cells are produced by the genes of that cell. So if I draw, this is the nucleus, it's got DNA inside the nucleus, this guy has the nucleus, it's got DNA inside the nucleus. If these guys are both B cells, and they're both coming from the same germ line, they're coming from the same, ancestry of cells, shouldn't they have the same DNA? Shouldn't they have the same DNA? And if they do have the same DNA, and I'll put a big question mark there, if they do have the same DNA, why are the proteins that they're constructing different? How do they change? And this is why I find B cells, and you'll see this is also true of T cells, to be fascinating, is in their development, in their hematopoiesis. I always have trouble pronouncing that word, but that's just the development of these lymphocytes. At one stage in their development, there's just a lot of shuffling of the portion of their DNA that codes for these parts of the protein. There's just a lot of shuffling that occurs. So in most of when we talk about DNA, we really want to preserve the information, not have a lot of shuffling. But when these lymphocytes, when these B cells are maturing at one stage of their maturation or of their development, there's intentional reshuffling of the DNA that codes for this part and this part. And that's what leads to all of the diversity in the variable portions on these membrane-bound immunoglobulins. And we're about to find out why there's that diversity. So there's tons of stuff that can infect your body. There's all sorts of viruses are mutating and evolving, and so are bacteria. You don't know what's going to enter your body. So what the immune system has done through B cells, and we'll also see it through T cells, it says, hey, let me just make a bunch of combinations of these things that can essentially bind to whatever I get to. So let's say that there's just some new virus. Let's say there's just some new virus that shows up, right? The world has never seen this virus before. And this B cell, it'll bump into this virus and this virus won't attach. And another B cell will bump into this virus and it won't attach. And maybe several thousands of B cells will bump into this virus and it won't attach. But since I have so many B cells having so many different combinations of these variable portions on these receptors, eventually one of these B cells is going to bond. Maybe it's this one. Maybe it's this one. And he's going to bond to part of the surface of this virus. It could also be to part of a surface of a new bacteria or part of a surface for some foreign protein. And the part of the surface that it binds on the bacteria, so maybe it binds on that part of that bacteria, this is called an epitope. Epitope. So once this guy binds to some foreign pathogen, and remember, the other B cells won't. Only the particular one that had the particular combination, one of the 10 to 10th. And actually, there aren't 10 to 10th combinations. During their development, they weed out all of the combinations that would bind to things that are inside of that are essentially that there shouldn't be an immune response to. So we could say self-responding combinations weeded out. So there actually aren't 10 to the 10th, 10 billion combinations of these, something smaller than that. You have to take out all the combinations that would have bound to your own cells. But there's still a super huge number of combinations that are very likely to bond at least to some part of some pathogen, of some virus or some bacteria. And as soon as one of these B cells binds, it says, hey guys, I'm the lucky guy who happens to fit exactly this brand new pathogen. He becomes activated. He becomes activated after binding to the new pathogen. And I'm going to go into more detail in the future. In order to really become activated, you normally need help from helper T cells, but I don't want to confuse you in this video. So in this case, I'm going to assume that activation can only occur, or that it just needs to respond. It just needs to essentially be triggered by binding with the pathogen. But we'll see in most cases you actually need the helper T cells as well. We'll discuss why that's important. It's kind of a fail-safe mechanism for your immune system. But once this guy gets activated, so this is the activated guy, he's going to start cloning himself. He's going to say, look, I'm the guy that can match this virus here. And so he's going to start cloning himself. He's going to start dividing and repeating himself. So so there's going to be multiple versions of this guy. Multiple versions of this guy. And not only are there multiple versions of that guy, so they have the receptor, the membrane, they have 10,000 of these. I'm only drawing one or two on each membrane. So they all start to replicate, and they also differentiate. Differentiate means they start taking particular roles. So there's two forms of differentiation. So they can go into, many hundreds or thousands of these are going to be produced. And then some are going to become memory cells, which are essentially just cells B cells that stick around a long time with a perfect receptor on them, with the perfect variable portion of their receptor on them. Let me draw a couple of them right there. So that is a memory cell. So that right there is a memory cell. So some will be memory cells, and they're going to be in higher quantities than they were originally. So if this guy invades our bodies 10 years in the future, they're going to have more of these guys around that are more likely to bump into them and start and get activated. And then some of them are going to turn to effector cells. And effector cells are generally cells that actually do something. So effector cells. Effector cells. And what the effector cells do is they turn into antibody. They turn into these effector B cells, or sometimes they're called plasma cells. They're going to turn into antibody factories. Antibody factories. And the antibodies they're going to produce are exactly this combination that they originally had being membrane bound. So they're just going to start producing these antibodies that we talk about with the exact. So then they're going to start spitting them out. They're going to start spitting out these antibodies. They're going to start spitting out tons and tons of these proteins that are uniquely able to bind to the new pathogen, this new thing in question. They're uniquely able to bind. So an activated effector cell will actually produce 2,000 antibodies a second. So you can imagine if you have a lot of these, you're going to have all of a sudden a lot of antibodies floating around in your body and going into the body tissues. And the value of that, and why this is a humoral system, is all of a sudden you have all of these viruses that are infecting your system. You have all of these viruses that are infecting your system. But now you're producing all of these antibodies. The effector cells are these factories. And so these specific antibodies will start bonding. So let me draw it like this. These specific antibodies, the specific antibodies will start bonding to these viruses. And that has a couple of values. I'll just draw them like that. I don't want to spend too much time drawing them. But that has a couple of values to it. One is it essentially tags them for pickup. Now, phagocytosis, this is called opsonization. When you have, let me write this down, ops again, going into the body tissues. And the value of that and why this is a humoral system is all of a sudden you have all of these viruses that are infecting your system. You have all of these viruses that are infecting your system. But now you're producing all of these antibodies. The effector cells are these factories. And so these specific antibodies will start bonding. So let me draw it like this. These specific antibodies, the specific antibodies will start bonding to these viruses. And that has a couple of values. I'll just draw them like that. I don't want to spend too much time drawing them. But that has a couple of values to it. One is it essentially tags them for pickup. Now, phagocytosis, this is called opsonization. When you have, let me write this down. Opsonization. When you tag molecules for pickup and you make them easier for phagocytes to eat them up, this is just called opsonization. These molecules are called opsonins. But it's essentially antibodies are attaching them, says, hey, phagocytes, this is going to make it easier. You should pick up these guys in particular. It also might make these viruses hard to function. Now you have this big thing hanging off the side of it. It might be harder for them to infiltrate cells. And then the other thing is that you have on each of these antibodies, you have two identical heavy chains. I'll draw it like that, and then two identical light chains. And then they have a very specific variable portion on each one. And each of these branches can bond to the epitope on a virus. So you can imagine what happens if this guy bonds to one epitope and this guy bonds to another virus. Then all of a sudden, these viruses are kind of glued together, and that's even more efficient. They're not going to be able to do what they normally do. They're not going to be able to enter cell membranes, and they're perfectly tagged. They've been opsonized so that phagocytes can come and eat them up. So we'll talk more about B cells in the future, but I just find it fascinating that there are that many combinations, and they have enough combinations to really recognize almost anything that can exist in the fluids of our body. But we haven't solved all of the problems yet. We haven't solved the problem of what happens when things actually infiltrate cells, or we have cancer cells. That how do we kill cells that have clearly gone astray?
It's 8 o'clock with no breaks. That's not bad.
In the very first immunology video where we talked about non-specific defense mechanisms, we said if we have some type of a pathogen, let's say it's a bacteria, that our phagocytes can recognize either proteins on the bacteria or maybe some other type of pathogen, some other marker on the pathogen. It doesn't know what type of pathogen it is, but that's enough for the phagocyte, and it'll engulf the pathogen. So this is the phagocyte. It somehow one of their receptors the very first immunology video where we talked about non-specific defense mechanisms, we said if we have some type of a pathogen, let's say it's a bacteria, that our phagocytes can recognize either proteins on the bacteria or maybe some other type of pathogen, some other marker on the pathogen, doesn't know what type of pathogen it is, but that's enough for the phagocyte, and it'll engulf the pathogen. So this is phagocyte, it somehow one of their receptors touches one of the proteins that's on the surface of this bacteria in this case, and says, gee, that's not a good thing to have around, and so it'll engulf it. So the membrane is just going to surround the pathogen, and this part is going to go in, it's going to kind of pinch in, and the pathogen is going to get surrounded and get engulfed, phagocytosed by the phagocyte. So the end product, we saw this on the first video where I talked about phagocytes, is we're going to end up, so then the membrane is going to be completely around, this is going to pinch in and bubble around this thing, and so we're going to have the pathogen sitting inside of the phagocyte. It's going to be surrounded by a membrane. That membrane is called a phagosome. And we learned there's different types of phagocytes. It could be a macrophage, could be a neutrophil, could be a dendritic cell, and we have it like this. And then we also saw that video that's not just done. The macrophage doesn't just digest this thing. That by itself is very useful. It got this bacteria out of the way. If this was a virus, it got that virus out of the way. It does even more than that. It takes that and then it lyses it, or it breaks it up. It doesn't have to just it breaks it up. There's several ways it can do it, but the bottom line is it processes it. We draw that. It'll break up the pathogen. We saw in that first video on phagocytes that will actually have a lysosome bond to it and dump all sorts of particles that are going to break up and cut up this bacteria, in this case, into its constituent molecules. Here we can draw that. So it's going to come here, and it's going to dump all of its contents and break it all up. And then the leftovers, some subset of the leftovers, particular chains of the peptides, and remember, proteins are long chains of amino acids, polypeptides are short chains. So you take short chains of those, and they're going to bond to special proteins. And this is essentially the topic of this video. So let's say they bond to that special protein, and then those proteins get transported or get to the membrane or the outer surface of the cell, and they present themselves along with a piece of the pathogen. So the end product, after we've phagocytosed this pathogen, is that we have the phagocyte will look something like this, and it will have these antigen presenting proteins, I guess we can call them, that had bound to parts of that original pathogen. Let me do it right here. So this right here. So it has a little bit of the original pathogen on it, and I do it in green right here. And these proteins right here, these are called, it's a fancy word, and I talked about it in the previous video, these are called major histocompatibility complexes, or MHC for short, MHCs. And when we're talking about phagocytes or macrophages or dendritic cells or particular cases of phagocytes, the major histocompatibility complexes that they present after they digested this molecule, this is an MHC class 2. So let me write this down. This is an MHC class 2 protein. It might seem like I'm really going into the minutia of what these proteins are. But we're going to see this is key for activating other parts of the immune system, especially the cell-mediated parts of the immune system. So this was the case with a macrophage or a dendrite. They engulf something, they chew it all up, and then the parts of the chewed up thing that they ate, they attach to these MHC2 proteins, and then these MHC proteins go to the top surface of the cell. The same thing. Or actually not this teaching. When we're talking about histocompatibility complexes, or MHC for short, MHCs. And when we're talking about phagocytes or macrophages or dendritic cells or particular cases of phagocytes, the major histocompatibility complexes that they present after they digested this molecule, this is an MHC class 2. So let me write this down. This is an MHC class 2 protein. It might seem like I'm really going into the minutia of what these proteins are. We're going to see this is key for activating other parts of the immune system, especially the cell-mediated parts of the immune system. So this was the case with a macrophage or a dendrite. They engulf something, they chew it all up, and then the parts of the chewed up thing that they ate, they attach to these MHC2 proteins, and then these MHC proteins go to the top surface of the cell. The same thing, or actually not quite the same thing, a very similar thing happens with B cells. Very similar. So if we have a B cell, a good color, B for blue. B stands for bursa, but it could stand for bone marrow just as well. Let's say we have a B cell, and it's got this membrane-bound antibody on it. Remember, it's very specific to that B cell. So all of the membrane-bound antibodies, all 10,000 or so of them on this B cell, they all express the same variable part. So this is a particular B cell. So remember, this was non-specific. When we talked about phagocytosis, these guys just said, hey, you're a bacteria, you're a virus. I don't know what kind you are. I'm just going to eat you up. You look shady. I'm going to eat you up. I don't know what type you are, whether I've seen you before. When we're talking about B cells, we're talking about the adaptive or the specific immune system. And so the variable ends of these membrane-bound antibodies are specific to certain parts of certain pathogens, to certain epitopes. Remember, epitopes were the parts of certain pathogens that these specific chains can recognize and bond to. So let's say that we're dealing with a virus in this situation. And let's say the virus just happens to bond to this B cell. Remember, there might be other, in fact, there definitely are tons of other B cells around, but their variable portions, the variable portions of all of these other B cells, and actually let me go for different colors so that the variable portions are all different. And that's what I always find amazing about B cells. That even though they come from the same genetic line, they have their genes have been shuffled around in their development so that they can produce billions of combinations of these proteins, but or the variable ends of their antibodies. So let's say we have some virus. Let me say it's a bacteria. When I did the other B cell example, I said we're dealing with viruses. But let's say some bacteria, some new bacteria, and just some part of its surface just happens to bind to only this B cell, because this B cell has just the right combination. So some part of his surface binds just to that B cell right there, and that part of the surface that binds, remember, that was called the epitope. That's that part of the pathogen that binds to our variable sequencer. It won't bind to this B cell or this B cell because they have different sequences here, but it binds to this B cell, and then that starts the activation process. We'll go into sometimes this by itself can get the B cell activated, but you normally need help from helper T cells, and we'll talk more about that. And we said once this happens, once you get activated or the activation process starts, this guy actually gets engulfed. And I didn't talk about that in the last video just because I didn't want to go into too much detail. So this whole thing gets engulfed by the B cell, and then when it gets activated, it proliferates itself, and you normally need the T cells there, and some part of them become plasma B cells, some part of them become memory B cells. So these are memory. Then you have the plasma B cells. Remember, the plasma B cells just say, gee, I've been activated. I'm just going to produce a ton of these antibodies. So the plasma B cell will just produce tons of these antibodies and start spitting them out so that they can attach to more and more of that pathogen and just mess them up in different ways. Either tag them, the green's the pathogen, tag them so that other macrophages or phagocytes eat them up, or tag two of them so you bundle them up so that they can operate properly, whatever. I'm not going to go into detail on that. That's when it gets activated. But the interesting thing is that the B cell will also do what the phagocytes do. The B cell will also take this guy in, into the cell, maybe he's initially attached to the antibody, and break him up, take pieces of this pathogen and attach it to MHC2 proteins, and then present them on the surface. So a B cell will also present the antigen. So this is also an MHC2 complex, a major histocompatibility complex. And just so you know, histo means tissue. So this is related to whether something is compatible with the tissue in your body. We'll talk more about that and how it relates to transplants and all of that. So this is also an MHC class 2. So in both cases, whether we're talking about B cells that recognize a very specific pathogen, and it can be a specific virus, a specific protein, a specific bacteria, or in the cases of phagocytes that just say, oh, you look shady, let me take you in. I don't know what type of bacteria or virus or protein you are. But in either case, they both engulf them, take pieces of them, cut them up, and present them on their surface in a complex with the major histocompatibility complexes. So cells that do this are called professional antigen presenting cells. Professional. This is what they do for a living, although they do other things. As we see, this guy, the phagocytes eat things. The B cells generate antibodies or memories so that they can later be activated to generate antibodies. But these are called professional antigen presenting cell. And the antigen in question is this little piece of of the actual thing that you're trying to track, that little piece that we got, the little actual part of the pathogen, that's what the antigen. So it's presenting the antigen. It's professional because it does this. It takes antigens from, or it takes pathogens from outside of our or in the fluid of our system, and then engulfs them, breaks them up, and then presents them. Now, there are also non-professional antigen presenting cells, and in fact, most cells are this. In fact, even these guys. So actually, I'm going to wait for the next video. I realize all of my videos are getting long. So you're probably thinking, gee, what are these? Okay, so what? These guys, in either case, engulf them, cut them up, present them. What is it good for? You'll see that these are these MHC2. These are what are recognized by helper T cells. So it'll all form part of the puzzle of how our immune system works. In the next video, I'll talk about MHC1 presenting cells, which is pretty much all body cells. In talking about the adaptive immune system, we've already seen that there's a couple of actors.
Holy crap, son. 20 minutes. Why take you so long, man?
Now,
Oh, you get the EMG stuff right here, huh? Oh, okay.
so let me write this down. Adaptive or specific immune system. Adaptive immune system. You have your humoral response. Humoral. So this is responding to things that are floating around in the fluids of the body and not necessarily things that have infiltrated your body cells. Then you have your cell-mediated response. Cell-mediated. And then in the humoral response, and we're talking about specific humoral response, this is where the B cells, the B lymphocytes are at their most active. And essentially what they do is you got a B cell here. It has a very specific antibody specific to just this B cell, not B cells in general. If this happens to be the one of the billions of B cells that happens to have the matching key, or maybe I should say the matching lock for the key that is the intruding pathogen, that pathogen will bind to that B cell. Maybe it's a virus, maybe it's a bacteria. And then the B cell will get activated. And we'll talk about in this video that the activation doesn't always happen. In fact, it usually doesn't happen just from this. But so far we said it gets activated. It goes into memory cells, memory B cells, which are essentially multiple versions of this original B cell, just saying, hey, let's have multiple versions of this because it tends to recognize this virus. So in the future, if we get this virus, those multiple versions, those memory cells are going to be there to have this interaction. This interaction is going to be more likely to happen in the future because I'm going to have more of this variety of B cell. And then you have effector cells. Effector cells, and these are essentially, so both of these are B cells. So this guy, once he gets activated, he proliferates, he keeps dividing and cloning himself. The memory cells just stick around waiting to be activated in the future. And I'm only drawing one of these membrane-bound antibodies, but there are actually 10,000 on the. I mean, I can draw a bunch of these. I don't have to just draw one. The memories just wait around in the future, but there's more of them now. So in the future, if we get this virus again, there's the reaction, this interaction is going to happen faster, and so they're going to get activated faster. And then the effector B cells essentially turn into antibody factories. So they just start, this antibody goes in, or it says, let me just produce, I've been activated, let me produce many, many more versions of that exact antibody. So they get spit out. Let me do that one a little wrong. So that exact antibody that can then be spit out to go disable or tag antigens, and not just any antigen, this antigen right here. And we also saw that the other thing that the B cell does is it becomes an antigen presenting cell. So what it does is, as soon as it recognizes this, it's had this interaction with an antigen that just matches the variable portion of its membrane-bound antibody. It endocytoses that. It brings that into itself. It's going to be membrane facilitated, so it just kind of pulls it in, chunks it up, and then presents a piece of that antibody on an MHC2 molecule. We saw that in the last video. So it cuts that up and presents a piece of it right there. And that's why we call it an antigen presenting cell. Now, in this video, we're going to talk about why we even have these MHC2 molecules. What are we presenting these antigens to? And so we're going to start talking about the cell mediated. And actually, even more than the cell mediated, we're going to talk about T cells. So we're going to talk about T cells. And I said in the first video, they're called T cells because they mature in the thymus. Mature in the thymus. And there are two types of T cells. And it's all very confusing. You have B cells and T cells, but then there are two types of T cells. You have helper T cells, and most people just write T with a lowercase or subscript H there. And then you have cytotoxic T cells, or T cells that kill other cells. Cytotoxic T cell, and that's T with a subscript C. Now, just so that you have a big overarching impression of what does what, B cells, when they are activated, they generate antibodies. That's at 30,000 feet, that's the best summary of what an activated B cell does. It actually generates antibodies. Those antibodies attach to viruses and bacteria and other types of pathogens and disables them, either tags them so that so that macrophages can go and eat them up, or just by throwing all of those antibodies onto the surface of the pathogen in question, it might disable the pathogens or essentially bundle them all together so that it'll be easier for macrophages to pick them up. But this is only effective for things that are floating around. Antibodies, free-floating antibodies, are only effective for things that are floating around. Cytotoxic T cells, which I'll cover in more detail in a future video, these actually attack cells that have been infiltrated. So this is attack, kill, infiltrated cells. And when I say infiltrated, it could be a cell that a virus has gone into or some bacteria has penetrated. And when I say infiltrated, it doesn't necessarily even mean something from the outside. It could even be a cancerous cell that shows itself to be abnormal in some way. And so the cytotoxic T cells will at least attempt to kill them. But what I want to focus on, so if you want, out of the three types of lymphocytes, these are, remember, everything we've been talking about is leukocytes, white blood cells, but lymphocytes are a subset of that. And these three are lymphocytes, and they're called that because they begin their development in the bone marrow. So this guy, and this guy actually do stuff. This guy generates antibodies to attach to pathogens floating around. This guy directly attacks cells that are broken in some way. They've either been infiltrated, they're abnormal, they're cancerous, who knows what. And I'll do a whole video on that. But that leads us to a very obvious question. What does this guy do? What does the helper T cell do if he doesn't directly interface either with pathogens or produce things that interface with pathogens, or if he himself doesn't go and directly kill cells? And the answer is that the helper T cell is kind of the alarm of the immune system. And on some level, it's almost the most important. So we talked already about, in the last video, about antigen presenting cells, that either when a macrophage or a dendritic cell takes things in, it cuts them up and presents it on its surface as these MHC2 proteins, or in complex with these MHC2 complexes or proteins. And so do B cells. B cells, they get attached, but B cells are more specific. Now, once something is presented, now the helper T cell can come into the picture. So this is let's do a dendritic cell, and I'm doing dendritic cells actually on purpose, because dendritic cells are actually the best cells at activating helper T cells. And we're going to talk about in a second what happens when a helper T cell gets activated. So let's say I have this dendritic cell. It's called dendritic, so it looks like it has dendrites on it. So the dendritic cell looks like that. So I have this dendritic cell here. It's a phagocyte. It's already consumed, let's say it's already consumed some type of bacteria or virus, and it's cut it up, and now it's presenting kind of the body parts of that virus on the MHC2 complex. So this is MHC2. It's kind of its way of saying, hey, I found this shady thing floating around in the body's tissues. Maybe someone ought to raise an alarm. Maybe this is part of some type of bigger thing going on, and some type of alarm bell has to be released. And that's what the helper T cell does. So let's say this guy, he's presented it. He says, I found this thing, I killed it. Here's a part of it. The helper T cell has a T cell receptor on it. So let's say this is the helper T cell right here. So this is a T helper, and it has a T cell receptor on it. And the T cell receptors bond to, and I'll be very particular here. So this is the T cell receptor, draw it like that. It's just a protein. But like the membrane-bound antibodies on B cells that every B cell, or almost every B cell, has a different version, different variable chain. That's also true of helper T cells. That just like the B cells, this has some variation in where it bounds. So this right here is going to be variable from one helper T cell to another. For example, I might have another helper T cell here that also has a T cell receptor, but the variable portion on that T cell receptor the variable portion on that T cell receptor is different than the variable portion on this T cell receptor. So this helper T cell will not bond or not bind to this dendritic cell or the MHC2 complex of this dendritic cell. Only this one would. And the mechanism of how you get this variation is very similar to the mechanism in how you get the variation of the antibodies in the B cells. During these helper T cells development, at some point, the genes that code for this part of this receptor get shuffled around, and they get shuffled around intentionally so that each T cell has a certain specificity, a combination of an MHC2 complex and a certain polypeptide, a certain part of a virus. So only this guy is going to be activated, not this guy. So this is why we call it the specific immune system. Now we say, what does the helper T cell do at that point? Well, at this point, once he says, hey, gee, I happen to be the one helper T cell that can bond to this guy, this antigen that's presented, it becomes activated. And I won't go into the details, but in general, dendritic cells are the best ones at activating it, especially a naive T cell. Let me use that word's important, naive. A naive helper T cell. In general, when we talk about a naive B cell or a naive helper T cell, these are cells that are non-memory, non-effector, that have never been touched by, they've never been activated. In the case of a B cell, they've never been activated by something binding to their membrane-bound antibody. Or a naive helper T cell is a non-effector, non-memory helper T cell that's never had anything bound to it. So if this guy is naive, and then he has a reaction with this antigen presenting cell, he becomes non-naive, he becomes activated. And when activated, two things happen. Well, just like with B cells, he proliferates many copies of himself, and some subset of those copies differentiate into effector cells, effector. An effector just means it does something. It does something now instead of saving the memory. Effector helper T cells, and then some subset of them become memory helper T cells after getting activated. Now, the memory T cells, just like memory B cells, now you have more copies of this. So in 10 years in the future, if something like this happens, this interaction is going to be more likely to happen. This guys have the same T cell receptor as their parent. This guy has the same T cell receptor as the parent. Actually, both of these guys do. This guy does as well. It's just that the memory T cells, or actually even the memory B cells, they last longer. They don't kill themselves. They'll last for years. So that if 10 years later, something like this starts presenting itself, you're going to have more of these guys around to bump into this guy so that you can raise the alarm bells. And this guy's also going to have, he's also going to have the same chain right there. So you're saying, fine, I have these memory cells. They're going to stick around so that this reaction can happen in the future. But I still haven't answered the question, what does the effector T cell do? What the effector T cell does is it raises the alarm. It starts releasing, let me draw an effector T cell, that same effector T cell up there, it's been activated. So this is an effector T cell. It has been activated. And it's, remember, this is very particular. Only this version of T cells, but once it got activated, it produced many copies of itself because it says, hey, I'm responding to a particular type of pathogen. So that this is a helper T cell. This is an effector. And what these do is they start releasing these molecules called cytokines. So they start releasing cytokines. So it starts releasing cytokines. Let me write that down, cytokines. And there are many different types of cytokines, and I'm not going to go into detail on all that. But what cytokines do is that they really raise the alarm. So if you have other activated lymphatic cells or other activated immunological cells, when the cytokines enter those cells, remember, cytokines are really just proteins. When the cytokines enter, or polypeptides, when they enter those cells, it makes them get in gear. It makes them multiply more often, or it makes them get more active in their immune response. So what this does, these cytokines you can do as chemical alarm bells, chemical or peptide alarm bells. It tells everyone to get in gear. So that's one role. And so you can see this is actually a very central role. And it'll tell both activated cytotoxic T cells to get in gear, which we haven't talked about yet. And it'll also tell B cells to keep proliferating. So when an activated B cell gets so this is an activated B cell. When it gets some of these cytokines that maybe come from a local helper T cell, it'll tell it, hey, divide more often, only if you've been activated already. And we'll talk more about why, it has to be that case, because you don't want all the B cells to be activated. And then the other thing that the effector T cell does, in the B cell discussion, I said, okay, if I have a B cell, and it has its membrane-bound antibody, and remember, this is a particular version. It has its particular variable chain right here. And then this guy binds to a pathogen. So this binds to a pathogen. Maybe it's a virus right there. Up to now, I've been saying that this guy is activated, and he's going to, once he binds to the pathogen, he'll take this in, and he'll take part of the pathogen. And cut it up and place it on an MHC2 molecule. And we've said then he'll be activated, he'll proliferate, and he'll differentiate into memory and effector B cells. But that's not quite true. This first stage happens. This guy bonds. This B cell happened to be specific to this virus, cuts up the virus up, puts parts of the virus on its surface, and presents parts of the antigen. But in most cases, this B cell isn't yet activated. It's just in kind of this, you can kind of view it as in this resting state, ready to be activated, but it hasn't started proliferating and differentiating into effector and memory molecules yet. And in order for that to happen, a helper T cell, an activated helper T cell that is also specific to this very same virus. So you can imagine someplace else in the cell, this virus was eaten by a dendritic cell. So I'll draw a dendritic cell like that. So this exact same virus, this exact same species of virus is eaten by that dendritic cell. And so the dendritic cell eats it up, it cuts it up, and then it presents it on an MH. It's an antigen presenting, so it presents it just like that. Then this will activate a very specific T cell, maybe that one. So then a very specific T cell will come and bump into it. Not just any T cell, the one with the right variable portion. So think about what's happening. The variable portion for this T cell, it connects to this part of the virus plus the MHC2, but it's really reacting to the same virus. It might be a different part. This little part that was cut off might be someplace inside the virus, while the epitope for the B cell might be someplace on the outside of the virus, but they're both specific to the same virus. Now, once this guy gets activated, and he starts producing memory and effector cells, or they're descended from him, one of those effector cells specific to this virus are needed to come bind to this guy. So then this guy can then go along and bump around and then eventually end up here. And then he is also specific to this virus. So this binding site right here is the same as this binding site, this combination of antigen plus MHC2. And so when this guy binds, so just like that, when he binds, and remember, this binding site is the same as this, and it only binds to this combination right here. This is what activates the B cell. In most cases, this is T-dependent activation, which is usually the case. There's sometimes all you need is this first thing, but in general, you need the first thing, and then you also need a T cell to come and activate it, and only then will the B cell get activated and start proliferating and dividing and differentiating itself and producing, when its effector cells will produce a lot of antibodies. And so there's a natural question, why do biological systems, or why do we have this double system? And at least my sense of it is it's a fail-safe mechanism. Because if every time a virus came and attached this guy just started going crazy and producing antibodies against this thing, there's some chance that maybe after development, this guy, this chain right here, or his genes for generating these chains, become specific not for foreign pathogens, but maybe they become specific for self molecules that are naturally produced within the body. And it's just a random mutation. But if he started going crazy for that, then his antibodies will start attacking molecules that are naturally in the body, and then that could really hurt. That's what causes autoimmune diseases, where your own immune cells start activating yourself. But if you have this double handshake system where this has to happen and this has to happen, the likelihood of both of these guys becoming, after they leave their development stage, becoming specific to a self protein or a self cell or a self molecule is very unlikely. So it kind of inhibits this guy just going wild, even if he has some type of mutation. Anyway, hopefully that explains a little bit of what helper T cells do. We'll talk a lot more about it. I know it can be a little bit confusing. In the next video, we'll talk about cytotoxic T cells. This is cytotoxic. I spelled it wrong in the first when I wrote it. Cytotoxic T cells. When we learned about antigen presenting cells, we learned that they can first digest something. Let me draw a dendritic cell right here, my best version of a dendritic cell. Maybe I should draw them simpler than that. A dendritic cell is a phagocyte, and it is an antigen presenting cell. So after it phagocytoses,
Let's take it a minute. I need a little bit of break. Just a little bit. We'll come back. We have a lot to do today. Yeah. I want to watch these videos too. I don't know. I gotta close some of these tabs out. Yeah. Okay. That'd be bad if I got locked out. Imagine I got locked out of the room. I don't have a key. That'd be pretty bad. Let's just finish this up, eh? I think we're almost done. Not really. We have to go till 12 again tonight. Not 12, but it's gonna be. But it's gonna be. I think last this was 1, right? I think this time should be at 12. Maybe just keep coming down. after. Yeah, I think maybe what we can do is we can do it after we finish, after we read the high unit guide, and then I'm most likely going to add the cards right after Khan Academy review. Let's do it. Yeah, today's a good day. Yeah, what a good day.
Cut it all up, and then it'll display. It'll present the antigen on its surface on a protein complex. Let me do it in a different color. So I'll do it in red. A protein complex here, and the part of the pathogen that it cut up, it'll put up right here. And we learned on the antigen presenting cell video that this complex right here was an MHC type 2 complex, where MHC stands for major histocompatibility complex, where histocompatibility just means tissue compatibility. And this was the case on antigen presenting cells. So even B cells did this. So let me draw a B cell. So a B cell, it has its membrane bound antibody, just like that. It actually has many, many thousands of these. I could keep drawing a bunch of them, just so you know there's not more than one. Maybe one of these get triggered or get attached to some type of virus or protein or bacteria floating around. And what it'll do is it'll take this in, and cut it up again, and cut it up and do the same thing as what the dendritic cell did. It'll cut up a part of this and present it on, so I made this in pink, so I'll make it pink right here, a little part of this virus. It'll present it on its surface in conjunction with an MHC2 complex. So once again, this is an MHC2 complex. So these professional antigen presenting cells that go out and take things out of the fluid, out of the humoral parts of our body, things just floating around, they'll take them in, they'll say this is bad, cut them up, and then present them on these MHC2. That's what we call them, professional antigen presenting cells. Now, it turns out that pretty much all cells in our bodies, so let me draw just a random cell in my body. When I say almost all cells, it's actually all nucleated cells. So all cells that have a nucleus in the human body. So the only cells in our human body that don't have nucleuses are red blood cells, which I find fascinating, so that they can have more space for storing hemoglobin. But all nucleated cells in our bodies have another major histocompatibility complex on it, and it's called an MHC1. MHC1, major histocompatibility type 1. And just so you know, these are also nucleated cells, so they're also going to have an MHC type 1 complex on them right here. Now, the interesting thing about the MHC type 1 complex is because it's on every cell in our human body, let me write that down, every nucleated cell, so pretty much everything but the red blood cells have an MHC1. This is where, if anything wacky is going on inside the cell, maybe the cell is cancerous and producing crazy proteins, maybe it's been infected with a virus, maybe some type of bacteria or some type of weird protein has gotten in here, any cell in the human body can cut those up, even if it's malfunctioning, and it will present them. So let's say the cell is cancerous. So this cell has all these wacky proteins that only cancer cells present that is not normal for a normal cell. That will be presented on the MHC1. Let's say that I have some other cell in my body. Let's say I have some other cell in my body that's a different type of cell. It's nucleated. Let's say it's been infected with a virus. So you have this virus. So it's turning into this virus factory. Those are trying to draw hexagons. Same thing. There are mechanisms in the cell that will take some of the proteins that make up those viruses and present them on the MHC1 complex. So it'll present parts of the proteins on the MHC1 complex. So in the case of MHC2, this is what triggered helper T cells to say, hey, you know what? I found something floating out here. Here's a little piece of it, Mr. Helper T cell. Why don't you bond to this and raise the alarm system? Now, the MHC1 system says, no, this isn't stuff floating around. I've been infected. I am cancerous. I'm going nuts. You better kill me. I'm a virus. I'm a virus-making machine. You better kill me. And that message goes to the cytotoxic T cells. That's really the topic of this video. So just to make sure you understand the difference. So T cells, they both have T cell receptors, but the helper T cells bond to MHC2 complexes. So if this was a helper T, let me scroll up a little bit. No, I have space up there. Let's say that this is a helper T cell right here. It would want to, and it's not all helper T cells will, only the ones that have the right combination, the right variable portion right here, that just perfectly bonds to this combination of an antigen and the MHC2 complex. This type of helper T cell, will bond here, get activated, and start differentiating. And the effector versions of them will start raising the alarms, and the memory versions of them will stick around in case this type of thing needs to happen again. With MHC1, instead of attracting a helper T cell, it will attract a cytotoxic T cell. Let me draw a cytotoxic T cell right here. So like helper T cells, the T cell receptor, it has a non-variable portion. It has a non-variable portion, but it also has a variable portion that is specific to this combination of antigen and MHC1. So maybe this cytotoxic T cell will be involved when this cell goes cancerous. This cytotoxic T cell would be of no use, or it won't bond to this one that was attracted to a virus. It's going to have to be another cytotoxic T cell that does that. And the mechanism where we get this variability in the helper T cells or the cytotoxic T cells, or you saw in the B cells on their membrane-bound antibodies, that all comes from when they're in their development stage. Once they start to, or in the maturation process, that the DNA that codes for these variable portions gets shuffled around intentionally. So normally we're always trying to preserve DNA information. Here it gets shuffled around. But anyway, once a cytotoxic T cell finds one of these guys on an MHC1, remember every nucleated cell in the body has an MHC1, then what it does is it gets activated. So let's say this guy says, hey, that looks shady, you need to die. So this guy gets activated, and just like all other activated cells, he starts to divide and differentiate. And he divides and he differentiates into memory, just in case you're going to need me again, just in case this type of cancer shows up again, and then also into effector T cells, which are the ones that do the killing. So this is an effector. So let's say one of these effectors, they'll also bind to cancerous molecules, cancerous cells, just like this one. So let's say I have another, let's say this cell has split, and there's another version of it right here. That's what cancer does. It divides aggressively. It's producing wacky proteins. It presents the wacky proteins on its MHC major histocompatibility type 1 complex. It displays the wacky proteins. And then one of these effector cytotoxic T cells will be attracted to it, just like that. And I'm not going into the details on what necessarily does the attractions and all the membrane-bound proteins. If you take an immunology class, you'll see more on that. So this is a cytotoxic T cell, and it essentially forces this cell to kill itself in a couple of different ways. One, it dumps, which are the ones that do the killing. So this is an effector. So let's say one of these effectors, they'll also bind to cancerous molecules, cancerous cells, just like this one. So let's say I have another, let's say this cell has split, and there's another version of it right here. That's what cancer does. It divides aggressively. It's producing wacky proteins. It presents the wacky proteins on its MHC major histocompatibility type 1 complex. It displays the wacky proteins. And then one of these effector cytotoxic T cells will be attracted to it, just like that. And I'm not going into the details on what necessarily does the attractions and all the membrane-bound proteins. If you take an immunology class, you'll see more on that. So this is a cytotoxic T cell, and it essentially forces this cell to kill itself in a couple of different ways. One, it actually can exocytose a bunch of proteins that they're called perforins that make little holes in the membrane of this cell. And then it has other proteins that it releases called granzymes that go in here and essentially start mechanisms that make the cell want to kill itself. So the big picture is, if you want to just take 20,000 feet, B cells are very effective at producing. So when a B cell gets activated, it produces antibodies that kill things that are floating around, right? Once a B cell gets activated, it starts producing a bunch of a bunch of proteins that they're called perforins that make little holes in the membrane of this cell. And then it has other proteins that it releases called granzymes that go in here and essentially start mechanisms that make the cell want to kill itself. So the big picture is, if you want to just take 20,000 feet, B cells are very effective at producing. So when a B cell gets activated, it produces antibodies that kill things that are floating around, right? Once a B cell gets activated, it starts producing a bunch of antibodies. These antibodies float around, and then they can bond up to viruses, make them ineffective, or essentially tag them for pickup from macrophages or dendritic cells or other types of phagocytes. While cytotoxic T cells, these are used to essentially kill cells that have gone awry. For example, a cancer cell that's presenting weird proteins, or once the virus has entered the cell, then the antibodies are really of no use. The antibodies aren't going to be able to get into those cells. And in that case, instead of cleaning up the virus itself, a cytotoxic T cell will come here and just kill this cell, because this cell is a virus factory. So you have to get it out of the way.
How does it kill it? Hey, that looks shady. You need to die. Oh my goodness. Hey, like say you need to die. And that's what I wanted to know as well, right?
Now that we've touched on all of the major players in the specific immune system, what I thought I would do in this video is kind of do a summary so it all fits together a little bit. So the first person or character we got exposed to was the B cell, which I'll always do in blue. So the B cell right like that. And what made that interesting is that every B cell has its own specific, or they have membrane-bound antibodies. But for each B cell, the membrane-bound antibodies on each specific B cell had its own variable portion. So this B cell, it'll be variable right like that, variable like that. And if I were to draw another B cell right here, I would draw the variable portion a little bit different. And this is why different actual B cells will respond to different antigens or different pathogens that have entered our system. And a B cell gets activated. It needs to get, let me write all this down actually. So if we're talking about a B cell to get activated, so let's talk about what happens when it gets activated or what needs to happen. So activation, it needs binding of the pathogen onto one of these membrane-bound antibodies. So binding of pathogen, we call it mutagen or antigen, but that's not all. I mean, sometimes that's all you need, but usually you also need to be stimulated by T cell. So helper T cell stimulation. You might say, where does the helper T cell stimulate this guy? Well, B cells were also antigen presenting cells, so he'll suck this guy in, break him out, and present him on an MHC2 complex. So that's an MHC. Let me do it someplace where I can draw a T cell coming in. Well, let's say this is an MHC2 complex. This guy gets cut up. Part of him gets presented right here. And then an activated helper T cell, whose variable portion of their T cell receptor specific to this could come along and activate this character. I'm not drawing that receptor well, but that right there is a helper T cell, and that is the B cell. Now, once it's activated, it starts differentiating, and it starts cloning itself, and it could either turn into effector cells. And this is true of B cells or T cells. Once they get activated, they keep cloning, and they either turn to effector cells or memory cells. Memory cells stick around a lot longer so that in the future, you're going to have many more of this version of B cell. So if you get the same antigen or pathogen in the future, the likelihood of it bumping into this type of B cell is going to be higher, so the response will occur faster. The effector B cells they essentially turn into antibody-making machines. So they'll say, gee, this antibody bonds to this antigen that we have in the system now. Let me just produce a ton of them. So it starts building up all of the cellular machinery, and it just starts producing antibodies like crazy. And I want to point out one thing that my wife pointed out to me when she overheard me making the last video, and she's a fellow in hematology, and a lot of hematology is immunology. So I definitely have to defer to her. She is the expert on this. And in the last video, I kind of, very hand-wavingly said, B cells, once they get activated, if they're the effector B cells, they produce antibodies. I want to be very clear. It is only the effector B cells that produce the antibodies, and the common term for them, if someone were to walk up to you and say, what cells in the body are producing antibodies, you wouldn't be wrong if you said effector B cells, but the common term that people expect to hear are plasma cells. Plasma cells and effector B cells are the same thing, but normally when they say what happens to a B cell when it starts producing antibodies, they then call it a plasma cell. They don't call it a B cell anymore. And I want to make that very clear because my wife's like, well, I have attendings that if they ask me what cell in the body produces antibodies, and if you said B cell, they would say, no, wrong, it's a plasma cell. Or if you said effector B cell, they wouldn't be happy. They wanted to hear plasma cell. This is the common term used in immunology and apparently rheumatology circles. Did I just say my wife is a hematologist? No, she's studying rheumatology. I get confused with all the pathology sometimes. Anyway, that's what the B cells do. And these antibodies can then go attach things and mess up viruses and antigen. Well, viruses are instances of antigens and bacteria and tag them for pickup by macrophages or other types of phagocytes. Those were the B cells. Then you have your T cells. And here I'm going to talk about T cells a little bit differently than I had in the last few videos, just to give a little bit more of a nuance. So there's two types of T cells. You might be able to say, hey, they're helper T cells and cytotoxic T cells. And you're not wrong. But what I'm going to do is do a slightly different differentiation just so that you what cell in the body produces antibodies. And if you said B cell, they would say, no, wrong, it's a plasma cell. Or if you said effector B cell, they wouldn't be happy. They wanted to hear plasma cell. This is the common term used in immunology and apparently rheumatology circles. Did I just say my wife is a hematologist? No, she's studying rheumatology. I get confused with all the pathology sometimes. Anyway, that's what the B cells do. And these antibodies can then go attach things and mess up viruses and antigen. Well, viruses are instances of antigens and bacteria and tag them for pickup by macrophages or other types of phagocytes. Those were the B cells. Then you have your T cells. And here I'm going to talk about T cells a little bit differently than I had in the last few videos, just to give a little bit more of a nuance. So there's two types of T cells. You might be able to say, hey, there are helper T cells and cytotoxic T cells. And you're not wrong. But what I'm going to do is do a slightly different differentiation just so that you are familiar with these terms. So there's two types of T cells, just like that. All T cells have T cell receptors. Let me draw the T cell receptors. T cell receptors, just like that. But they also have these other proteins on them. And some of them have these proteins called, I'll draw it like that, called CD4 proteins. And some of them have, I'll do it in a different color, and some of them have what's called CD8. CD8. So this one right here would be called a CD8 positive T cell. It has the CD8 proteins on it. And this would be called a CD4 positive T cell. I've never used these words before. You're like, D, where are these coming from? Now, the CD4 receptor is the thing that wants to bind. This is the thing that helps to go to the MHC2 complexes. So most CD4 T cells are helper T cells. So this is, most of the time, and I want to make it very clear, immunology is this field, people are discovering things on a regular basis. So people are still understanding these things, and there's all sorts of special cases. But usually when people talk about CD4 positive T cells, they're talking about helper T cells. So this is normally going to be a helper T cell, or you could call it a T helper, just like that. Likewise, the CD8 proteins, these are attracted to the MHC1 complex. These are attracted to MHC1. You can kind of say this is what brings them to the cells that have the cancer that have expressed antigens on their MHC1 complex. So most of the time, CD8 positive T cells are cytotoxic. Cytotoxic T cell. And oftentimes before a cell gets activated, they just describe it as a CD4 T cell or CD8 T cell. And then after it becomes, for example, in a cytotoxic T cell, after it becomes activated and starts wanting to kill things, then maybe you call it cytotoxic. But this is all wordplay. I think you get the general idea. But just to remember what they do, this guy, we just said, he'll bond. He wants to bond to the MHC complex. So you have MHC complex plus presenting some antigen. This is MHC1 right here. We learned in the last video, every nucleated cell in the body expresses an MHC1 complex. So this is the case where something wacky is happening inside the cell. Maybe a virus has infected it. Maybe it's cancerous. It needs to die. Otherwise, it's going to keep producing proteins or keep producing viruses if it's infected by a virus, or otherwise it's going to keep dividing if it's a cancer and infect the rest of the body. So the CD8 kills infected cells, I'll just say bad cells. I don't know if you can cancer release it to infection, kills bad cells that are, if you don't kill them, they're going to keep producing viruses or keep splitting and spreading the cancer. While T cells, they're attracted to professional antigen presenting cells. So, and I always do a dendritic cell right here because they're also the best antigen presenting cells. And they have MH2 complexes, and it's digested some antigen and it presents it right there. And then that activates the helper T cell. And then when the helper T cell, all of these guys, once they're activated, they all go into they all start differentiating into effector and memory cells. An effector helper T cell, it does a couple of things. So if we're talking about helper T cell, it can activate B cells, and it also releases cytokines. Let's say this guy gets activated, he'll also start releasing these chemicals, which are really those alarm bells that tell other people to really get in gear. Maybe, B cells and cytotoxic T cells start proliferating more rigorously. Actually, part of the T cell the cytotoxic T cell activation can be assisted or kind of be given a boost by these cytokines, so by these alarm bells. So this guy is the alarm ringer. Alarm. While the CD8 cells or the cytotoxic T cells, when in their effector mode, they kill cells. they kill cells. And of course, in the memory mode, there's just a bunch of copies of these originals around that are ready and more than there originally were, so that in the future, if something like this or something like this occurs, they're going to be activated faster because they're going to be bumped into faster. So hopefully that clears up a little bit, and I introduced a little bit more top terminology. But I really want to stress my wife's point, because she said, hey, you don't want people out there saying B cells produce antibodies, even though it is effector of B cells, once activated B cells that are differentiated into effector.
In this video, I'd like to give an overview of how the B and T cells in your body are prepared to fight infections. And so let's start with where B cells come from. So where do B cells come from? They come from the bone marrow. So here's a bone, and inside it we have some bone marrow, and it's from there that B cells come from. But the process by which the B cells are prepared in the bone marrow is actually very interesting and unique in terms of the human body. Because, well, like many other places in the body, the B cells divide and form millions of descendants. And that sort of is how your whole body grows and comes to be as big as it is. But for B cells, there's something very different from other parts of your body, and that is that each daughter cell that grows up in the bone marrow is different from its parent, and it's also different from all of its siblings. So just to remind you, B cells have these receptors that later on will be their antibodies, but each daughter cell of this original B cell will have a different receptor. And that's because in the process of being made, these daughter B cells will have a slight shuffling, a slight changing in their DNA, but specifically for this receptor. And so by the time they're created, they have a unique receptor that will identify a unique criminal, a unique bacterium or virus or something in the body. But that receptor will be useless for all the other criminals in the body. So it's very specific, but very powerful if it finds the thing that it wants to kill. So this is happening in the bone marrow. And basically the same exact thing is happening for T cells, except it's not happening in the bone marrow. That's happening here. Do you know what this is called? This is called the thymus, and I'm drawing it a little bit bigger than I should maybe, but that way you can see it clearly. The thymus has two lobes or two parts, and it's located right behind your sternum. So T cells come from the thymus, and very similar to B cells, they have a unique receptor. It's not quite the same, and it doesn't become an antibody later on. They have a unique receptor, and while these T cells are dividing and creating millions of daughter T cells, each of these daughter T cells will have its own unique receptor that will observe two parts, and it's located right behind your sternum. So T cells come from the thymus, and very similar to B cells, they have a unique receptor. It's not quite the same, and it doesn't become an antibody later on. They have a unique receptor, and while these T cells are dividing and creating millions of daughter T cells, each of these daughter T cells will have its own unique receptor that will also identify a unique criminal or bacterium in the human body. And we remember that these receptors actually bind antigens, so pieces of those criminals that are presented to them by antigen-presenting cells, and we'll revisit that in a second. So all this is occurring in the thymus. And so this is step one of the process, and it's unique to the human body because we end up with all these actually literally genetically different B and T cells that are very specific to things which may or may not even exist out there because they're really created at random. Now, the next thing that happens is that these B cells and T cells, once they're ready, are going to migrate to a lymph node. And you have lymph nodes all over your body, about 600 of them. But let's say that they go to one here in your armpit. They're going to migrate to that lymph node, for example, and that is where they're going to wait for their special criminal that they can react to. So they're going to go over there to that lymph node. And let's draw that down here, maybe significantly bigger. So here's the lymph node, and you're going to have your B and T cells, all the different kinds of them, lying in wait there. Migrated here to all the different lymph nodes of the body. And now these guys are ready. And so the next step is really infection. So let's look at a tissue in your body with some cells. These are cells. Let's say it's over there. So it's not too far from that lymph node there. And let's say it gets infected. So here are some bacteria which are causing damage. And one of the things we have out here is these cells which are part of the immune system, which are called dendritic cells. Dendritic cells. And these cells can eat some of these bacteria. So they can actually kill some of those bacteria. They can ingest them. But it's actually not quite to the point that they can defeat this infection. And that's not really their purpose. And their purpose is actually more to present these bacteria to the B and T cells that we've been talking about. And so the way that they do that is that they come here into these lymphatic vessels, which start here in the tissue, and they go up to the lymph node where they're going to present these bacteria to these cells. And at the same time, some of the bacteria might also get swept in there and might go into the lymph node. And so you get some of them out here. You get some bacteria and maybe just some pieces of bacteria that got broken down. And you also have your dendritic cells here, which are antigen-presenting cells, which means that they have this protein that we've talked about called MHC, on which they present pieces of those bacteria that they have ingested. Now, these T cells here are unable to identify these bacteria when they're just freely floating. They need this antigen-presenting cell to show this piece of bacteria to them. And only the T cells, which can recognize this very specific pathogen, are going to see it and bind to it and react to it. So in this case, it's this guy. He's going to bind to that little piece and recognize that his invader, the one guy that he's been created to kill, is somewhere in the body. Meanwhile, these B cells are made to react directly to these bacteria without this antigen-presenting cell. And so they are, the specific one that is specific to this bacterium is going to see it, bind to it, and also be alerted. However, all these other guys, they're not going to know that anything's going on because they can't bind to any of these pathogens that were brought into the lymph node. And so this right here is step 2, when the infection occurs and the right B and T cells are alerted. And what are they going to do now? Well, now they're going to activate. They're going to jump into action because finally they're needed. And now these B and T cells are going to start replicating like crazy. So here are the two lucky winners who got selected. And they're going to say, well, now that we know that we are useful for fighting this infection, we better replicate like crazy because right now there's only 2 of us and that's not nearly enough. So they're going to start replicating, and each of their descendants, unlike this kind of replication, now each of their descendants is going to have the same receptor. It's going to have the same receptor. If it had a different receptor, this whole process would be useless because it wouldn't be able to fight the specific infection that we've just identified. And I should mention that not all these guys are going to do the same thing. So maybe most of them will become effector cells, which means that they're going to be ready to fight this infection right now. But some of them are going to become memory cells, which means that they're not going to actually fight right now. They're going to wait until the next time that this pathogen comes around. And the reason why we create these guys is because if we get this bacteria once, there's a high chance that we're going to get it again. Because really, you create billions and billions of these different receptors, but only a very small subset of them will actually end up being pathogens that you see. And so this is step 3 of our process, which has finally resulted in an army of B and T cells ready to fight this specific invader. And so that's what they're going to do now. The B cells are going to pump out antibodies to attack this bacterium. And the T cells, some of them are going to stay in the lymph node, and some of them are going to travel out to this site to join the battle. And so this whole process can actually be called clonal selection. Clonal selection, which is a name for basically for the theory of this process when it first came out. So this is clonal selection. And the reason it's called clonal selection is because you're going to select the B and T cells that you need, and then you're going to start cloning them like crazy into this army. And the process involves all these very unique, specific B and T cells coming from the bone marrow and thymus to the lymph node, where they're going to be met by these bacteria that are infecting your skin, that are going to travel through the lymph node, through the lymphatic vessels to the lymph node. And so they'll meet there, and then the armies will be prepared.
Prepare the troops! Prepare the troops!
We've all
Sheesh.
had cuts on our finger or wherever else on our body, and immediately that part of the body, they get a little bit of redness, a little bit of swelling, some heat maybe, and of course there'll be some pain associated going on there. And in general, this set of symptoms that we experience, these are known as the inflammatory response, or you might say that there's some inflammation going on there. And people have known about this, I guess, ever since really people have been having cuts. They probably, I think probably with modern medicine, have people been a little bit more particular about actually classifying the symptoms. But this isn't anything new for someone to say that there's some type of inflammation going on or some type of inflammatory response. But what I want to do in this video is understand what's causing these, I guess we call them macro symptoms, what's happening at a cellular level. Because really, the inflammatory response is essentially the initial field of battle of our immune system. You know, our first line of defense is our skin or the fluids on the outside of our skin or the mucous membranes. But the inflammatory response is what happens when something gets beyond that. We get punctured with a nail, or there's some type of virus or bacteria get beyond our skin or the mucus that surrounds our membranes. So this is the battle. This is the field of battle. Field of battle, especially the initial field of battle. So let's set up an immune battle so we can see exactly what's going on with the inflammatory response. And I want to be very clear. Immunology, people, it's still a very understood field. It's an area of active research. People are still discovering the mechanisms, and it's hugely complex. I'm sure we'll probably be studying this for a long time to come. So what I'm going to talk about is just the overview, just so you know the general actors, and you know in general what's causing the redness, the swelling, the heat, and the pain. So let me draw some skin cells. So let's say that these are skin cells. These are a gross oversimplification of everything, but it's really just to give an idea of what is going on. So I'm going to do a cross section. So those are some skin cells in there. And then I'm going to, so this is the outside world right here. Outside world. And amongst those skin cells, I'll do some other cells, and we'll talk about what they do. I won't go into huge detail about them. Let's call this cell right here, let's call this a mast cell. I'll draw a few more mast cells, maybe another one right there. That's a mast cell. And if you remember from the videos on phagocytic action or phagocytes, you remember there's one type called dendritic cells, and they tend to hang out near our skin. They kind of hang out near areas that might interface with the outside world. So I'll draw a couple of dendritic cells. And these were also the ones that were really good at activating, so this is dendritic, at activating helper T cells. Dendritic, maybe I'll do one more dendritic cell right here. Right there, that's a dendritic cell. And they're called dendritic cells. They have no relation to the nervous system. They just look like they have dendrites on them, and that's why they call them dendritic. But they're really phagocytes, and they tend to be near external interfaces, and they phagocyte host particles, and they're good at presenting them to helper T cells so that they can get activated and ring the alarm bell, so to speak. So this is just a normal, functioning, happy skin. So that's the outside. Over here, this is the interstitial fluid. Interstitial fluid. That's just a fancy word for the fluid that cells are kind of being surrounded by, or that bathes cells. You know, the cells aren't all directly connected to the circulatory system. The oxygen goes from the circulatory system to the interstitial fluid and eventually finds its way into cells. So it's not, you know, everything is directly connected to capillaries, but capillaries play a big role in our circulatory system. So let me draw that. Instead of just drawing them as tubes, I'm actually going to draw the cells of our circulatory system. So let's say that this down here, these are the endothelial cells of our capillaries. These are literally the cells that make up the walls of our capillaries. Right there, that's one side, and let me draw the bottom side right here. These are the actual cells, and of course, this is a cross section. If I were to, you know, I would draw it as a tube somehow. It's not like it's a sandwich. It's actually a tube. Obviously, everything is in cross section. So these are endothelial, spelling is the hardest part. Endothelial capillary, or may I say capillary endothelial cells. And of course, right in here, this is what we have our blood flowing, and we'll have red blood cells in here. Maybe I'll draw a couple of red blood cells in here. These are red blood cells. Maybe they're flowing in that direction. On this side, they're oxygenated. This would be arteries, and then they become veins as the red blood cells lose their oxygen. And of course, you might have circulating white blood cells inside of your, obviously in much lower quantity than your red blood cells, but just to show that they're circulating and they're moving with your circulatory system being pumped by the heart. Now, under normal circumstances, there is an exchange of, obviously, of gases between what's going on in our circulatory system and the interstitial fluid. And there's also a mild exchange of some cells and proteins. What we're going to see now is what happens when we have an intruder. So let's say someone takes a nail, dips it in some cow manure, and then pokes you with that nail. So let's see what happens. So someone pokes with them, it's a big nail, these cells are. So let's say someone pokes you with this nail dipped in cow manure. So it's got all sorts of nasty stuff on it, pathogens on it. Probably has a bunch of bacteria sitting in the cow manure. I didn't pick cow manure at random. It's probably a good source of bacteria. So as soon as it pierces the first line of defense, as soon as it pierces your skin, a couple of things are going to happen. A lot of these bacteria are immediately going to start floating around in your interstitial fluid. The cells that it came in contact with, it pierced them. It probably killed some cells. It's also going to damage some of these skin cells. And those skin cells are immediately going to start releasing chemicals, the ones that are still in a position to do so. They're going to start releasing chemicals that are essentially chemical messengers that move through at first locally in the interstitial fluid, and says something is going on. Something has happened to me. And these are called chemokines. Chemokines. And chemokines are just a very general word, really, for small molecules or small proteins that cells release as a kind of signaling mechanism. Chemo for chemical, kine for kinetic, for moving. These are messengers. They move. So these chemokines get released. And this is all tremendously complicated. So I'm doing a very high level. There are many, many types of chemokines. And also you have these mast cells here. These mast cells can be activated by direct contact, maybe with a rusty nail. It could be from the chemokines released by some of these cells up here. It could be from some of the molecules released by the actual bacteria. These bacteria are also releasing, different byproducts as they enter the body. And any of the above can activate the mast cells, and mast cells release histamine. Histamine. So the mast cells release histamine. So I'll do that in that color. Histamine. So you can already appreciate I'm doing a high-level overview that already is kind of complicated, but I think you get the sense of what's going on. And if the word histamine sounds vaguely familiar, it's probably because you've taken an antihistamine sometime, probably in the last several months, especially during cold season. Histamine is kind of one of the main actors in the inflammatory response. And when you have a cold and a runny nose and stuffy nose and all of those type of things, those are all byproducts of the inflammatory response. And antihistamines essentially try to shut down that inflammatory response, so some of those symptoms disappear. But it begs the question of, is that necessarily always a good thing? Because as I'm going to talk about in this video, this is the first line of defense. This is the first part of the battle of our immune system. But anyway, so the histamine, one of the things that the histamine does is it goes to the endothelial cells that line your capillaries, and it causes them to separate away from each other and make the actual capillaries larger. It's called vasodilation. So let me erase some of these and make them get larger and make the area larger. So let me erase these. Let's say that histamine, they've all been activated with a little bit of histamine. So let me go back. Okay, so then the histamine has come in. So now these guys get further apart. They get further apart, and the actual capillary becomes larger. So there you go. And then on this side, it's also going to happen. So this is where you get a lot of your swelling, because all of a sudden the capillary is larger, more fluid. Actually, it gets smaller further down. So it encourages the fluid to collect right around here. So this is called vasodilation. Just another fancy word for saying your capillaries are getting dilated, they're getting larger, fluid is filling them up. And not only are things starting to collect here, more and more red blood cells are collecting here. Obviously there's a lot of fluid here, the white blood cells, but also the capillary walls are becoming more porous. All of a sudden, things that couldn't get through them are going to have a much easier time getting through them. And one of those things they're going to have an easier time getting through them. And once again, remember, all this other stuff is going on. You have these histamines that are, being dumped on these endothelial cells, and maybe some of it's getting into the serum. You have these chemokines. You have these chemokines being released locally from this area of damage. You have the actual things being, the green was the color of the molecules being released by the viruses. You have the chemokines, which are in blue. They're all being released here. And your first line, and so the first responders, which we talked about, the phagocytes, and in particular the neutrophils, which are the most abundant of the phagocytes, a subclass of white blood cells, they're attracted to these chemicals. They're attracted. They want to move in the direction that there's more of these chemicals. And now that the space between these capillary cells have gotten further apart, they can get through. So what they actually do is, let's say that this right here is a neutrophil. This right here is a neutrophil. They start kind of rolling along the wall right here. That's called marginization. They roll along this wall, and eventually, so they kind of stick to the wall, they adhere to the wall, and then eventually they squeeze through these gaps in the capillary wall. This is called diapedesis or extravasation. Sometimes it's called emigration. It's all fancy words, but essentially it's just squeezing through the wall. So that's the neutrophil right there. And then, of course, because of the vasodilation, this is where the neutrophils will be getting dumped in, and this is exactly where they're needed. So these neutrophils are going to be here, and then they're going to do what they do. They're going to phagocytose some of these bacteria and start eating up, and maybe even some damaged cells up here. And so that's what you want to happen. That's why I said this is the field of battle. At the same time, your dendritic cells, other phagocytes, they will eat up the viruses, and then they'll present them on their surfaces. And it's not just neutrophils that are coming in, because this is kind of an area of congestion in your all of the fluid is coming here. You'll also have B cells and T cells. They'll also make their way. They'll also experience marginization, where they roll up against the sides of the capillary, and then diapedesis or extravasation where they go through and then they'll be activated and they can actually do the specific immune system. So the whole point here is I wanted to show you, and this is why I delayed the whole video on the inflammatory response, because it isn't just some one type of simple thing. It's actually the field of battle where all of the actors come into play, even the first line of defense of your skin, and then all of the actors, the non-specific reactions of inflammatory response is normally categorized as non-specific because it's going to happen no matter what comes. But you have the non-specific actors like the neutrophils, you have your specific actors like your B cells and T cells. And you also have the non-specific complement system. And I'm not going to go into detail here, but you actually have proteins that are flowing in your blood plasma that are normally in an inactive state. But when the inflammatory response occurs, these proteins, they get essentially activated. And sometimes, and this is all not 100% well understood, they become activated, they get cleaved up, and then the cleaved up versions of those proteins are really good at, in a very non-specific way, helping to kill off at least some of what's getting, maybe the bacteria in this case. So this right here, this is the complement system, which is really just a set of proteins that always just floats around, and they are good kind of first line of non-specific fight against some type of invading pathogen. So hopefully this gives you a good sense of what is going on in the inflammatory response. And as you can imagine, you have all this fluid coming here, all of this blood is collecting here. You have all of this fluid coming into, so not just cells that are going from our capillaries into our interstitial fluid, you'll actually have fluid going in, and that fluid that's going in is called exudate. So this whole thing becomes swelling and red and engorged, and that's why you see on kind of a very macro level these type of symptoms. Anyway, hopefully you found that useful.
So the purpose of the immune system is to fight things. It's to fight bacteria and viruses and other things that you don't want in your body. So that's what it does. And a question we can ask is, how does the immune system know not to attack your own body? And that might seem like a strange question or an obvious question, but it's actually not obvious, the answer to it. And so in this video, we're going to go into why it's not obvious and then how the body actually does prevent its immune system from attacking itself. So another way of saying this is, how does the body distinguish self versus non-self? And by that we mean, how does it tell the difference between your own body, your own proteins, your own cells, and foreign proteins or cells or things that shouldn't be in your body? So how does it know to attack these and not to attack these? So to find out why the question itself is not obvious, let's go back to B cells and talk about B cells. So here we have a B cell, and here's its nucleus with some DNA. And the most important part of the B cell that we care most about is its B cell receptor, which later, if it becomes active, can be released as an antibody. Now this B cell receptor is what's going to bind to foreign pieces of foreign bacteria or viruses, and these antibodies are going to bind to those things and help your body get rid of them. And the important thing to remember about B cells is that these antibodies or these B cell receptors are coded in the DNA of the B cell, but that they're different for every B cell. So every B cell has a unique set of antibodies and B cell receptors that it makes. And so we'll give them slightly different colors to make that obvious. Here's one with slightly different DNA and a slightly different B cell receptor. And the really critical point to remember is that these B cell receptors that will become antibodies are generated at random. So your body kind of shuffles the DNA here and creates a unique B cell receptor and antibody for each B cell. And it's precisely that fact, which is that they're created at random, which means that your body is in danger of creating B cell receptors and antibodies that can react to your own body. Because while this guy might be good, while he might react to, let's say, a bacteria out here that you want to get rid of, so while that's good, this guy might easily create a B cell receptor and latent antibody that can react to something you don't want it to react to. So for example, let's say this is an important protein in your body, maybe it's insulin. You would not want a B cell to be created which will react to insulin, because then maybe it'll start creating antibodies that bind all the insulin in your blood. And if you don't know what insulin does, don't worry about it. But by binding to insulin, it'll prevent insulin from doing its function, which is very important. So how can you keep your body from making B cells that would react to yourself? In fact, there's no way to do it, because, as I said, this process of creating different B cell receptors and antibodies is totally random. So there's no way to keep your body from making B cell receptors or B cells that will react to yourself. So what does that mean? That means that you're going to make them, but you need to find a way to figure out which ones are reacting to you and to get rid of them. So you need to figure out a way to kill the ones you don't want. And by the way, we're talking about B cells here, B cells, but everything we're saying is equally applicable to T cells. So we draw one here. T cells, it's equally applicable because T cells also have a T cell receptor that's generated at random. You only cells are things that shouldn't be in your body. So how does it know to attack these and not to attack these? So to find out why the question itself is not obvious, let's go back to B cells and talk about B cells. So here we have a B cell, and here's its nucleus with some DNA. And the most important part of the B cell that we care most about is its B cell receptor, which later, if it becomes active, can be released as an antibody. Now, this B cell receptor is what's going to bind to foreign pieces of foreign bacteria or viruses, and these antibodies are going to bind to those things and help your body get rid of them. And the important thing to remember about B cells is that these antibodies or these B cell receptors are coded in the DNA of the B cell, but that they're different for every B cell. So every B cell has a unique set of antibodies and B cell receptors that it makes. And so we'll give them slightly different colors to make that obvious. Here's one with slightly different DNA and a slightly different B cell receptor. And the really critical point to remember is that these B cell receptors that will become antibodies are generated at random. So your body kind of shuffles the DNA here and creates a unique B cell receptor and antibody for each B cell. And it's precisely that fact, which is that they're created at random, which means that your body is in danger of creating B cell receptors and antibodies that can react to your own body. Because while this guy might be good, while he might react to, let's say, a bacteria out here that you want to get rid of, so while that's good, this guy might easily create a B cell receptor and later an antibody that can react to something you don't want it to react to. So for example, let's say this is an important protein in your body, maybe it's insulin. You would not want a B cell to be created which will react to insulin, because then maybe it'll start creating antibodies that bind all the insulin in your blood. And if you don't know what insulin does, don't worry about it. But by binding to insulin, it'll prevent insulin from doing its function, which is very important. So how can you keep your body from making B cells that would react to yourself? In fact, there's no way to do it, because, as I said, this process of creating different B cell receptors and antibodies is totally random. So there's no way to keep your body from making B cell receptors or B cells that will react to yourself. So what does that mean? That means that you're going to make them, but you need to find a way to figure out which ones are reacting to you and to get rid of them. So you need to figure out a way to kill the ones you don't want. And by the way, we're talking about B cells here, B cells, but everything we're saying is equally applicable to T cells. So we draw one here. T cells, it's equally applicable because T cells also have a T cell receptor that's generated at random. And you only want that T cell receptor to react to foreign things, to non-self things, and not to self things. So the processes we're going to talk about are equally true, maybe even in some cases more so true for T cells than B cells. So let's go to the bone marrow to figure out how this process works. And we're going to the bone marrow because that's where B cells come from. It's where they get their unique antibody or B cell receptor. They get that by changing their DNA a tiny little bit, by shuffling pieces around. And so let's look at a couple of these B cells, which are still young. They haven't yet been allowed out of the bone marrow. They haven't been vetted to see if they should be allowed out. And each one has its unique receptor. So let's draw a few of those receptors here. And let's say that one of these guys reacts to self. One of them reacts to some protein in your own body that you don't want it to. And again, that just happened at random because you're creating these receptors really at random. So let's say that the guy that we're going to want to get rid of is this one, because he reacts to, let's say it's insulin again, look up there. Really, we should have drawn insulin in yellow to show that it goes with this receptor. So let's say this guy reacts to insulin. So how can you figure out that this guy reacts to self? And the answer is actually quite simple. The answer is that you just need to keep around the various proteins that your body uses. You need to keep them around in the bone marrow while these B cells are being vetted. And so, for example, here you'll have a little insulin, very small amount, but it'll be there. You'll have a little bit of, some other protein, let's say maybe hemoglobin. You'll have some other protein here. You'll have yet another protein over here. And so all these proteins will be around. And so what your body does at this stage in development is it says, whatever B cell binds to something with its B cell receptor in the bone marrow will be killed. And so this B cell right here that recognizes this insulin protein, the fact that it recognizes it means that it'll bind, and that'll cause a little bit of a chemical change in the B cell or something. And one thing will lead to another, and the whole system will be programmed so that as a result, this guy will die. So every B cell that recognizes self, if it sees that self molecule in the bone marrow, it will be killed. And this works because your bone marrow will have most of the abundant proteins in your body. They'll be present there so that you can make sure that you weed out all the B cells that react to self. Now, what happens after this step is that these guys who have been vetted, they can proceed onwards to maybe a lymph node somewhere where they can begin to actually be active now that they've sort of, gone through basic training here in the bone marrow. And you might ask yourself, well, what about here? What about here when one of these B cells that doesn't react to self, what about when it interacts with a bacteria that you actually want it to fight? Is the same thing going to happen? Is it going to die just because it recognizes the molecule that it's made to bind to? And the answer is obviously not. You don't want this guy to die because you need him, because you want to fight this bacteria. And so the reason why he doesn't die is because, well, we're in a different environment. There are different rules, there are different other cells around, and this B cell has matured and become different. So the rules are different, and he's not going to die. So this weeding out of B cells that react to self proteins is sort of the first of two mechanisms that I'd like to talk about that the body uses to not react to self. And actually the same exact thing happens for T cells, except it doesn't happen in the bone marrow, it happens in the thymus because that's where T cells mature. So in the thymus, we have really the identical process where T cells differentiate and each one has a unique receptor, and the ones that react to self in the thymus too strongly are killed. But it's not a foolproof method, or else we wouldn't need step 2. Every once in a while, a B cell will get out there. A B cell will escape, which reacts to self. And it's just because every process has its mistakes, and maybe you don't have every single protein here in the bone marrow in enough abundance to find B cells that react to a protein of your own body. So let's say this is a B cell that escapes the bone marrow even though it reacts to self. What's going to happen now? Well, it's going to find that protein that it was sort of made at random to react with. So it's going to find that protein that your body makes and that your body needs, and it's going to bind to it. What's it going to do now? Well, if you remember, it's now going to take that protein, ingest it, break it up into little pieces, and then present it on an MHC2 molecule. MHC2 molecule. And if you recall, it'll just present a small piece of that protein on the MHC2 molecule. Maybe it'll present a different piece of protein on a different MHC2 molecule over here, something like that. And the reason it does this is because it needs a T cell to come along. So here's a T cell. It needs a T cell to come along that will recognize that same piece that is put there on its surface. And it needs that in order to activate. So it's going to sit there and wait for this T cell to come along who has the perfect receptor. And so here's that T cell. And they're going to interact, and they're going to have some kind of intracellular kiss that's going to finally allow this B cell to activate. So usually without the T cell coming and recognizing the antigen that the B cell reacts to, the B cell cannot activate. It needs this T cell to recognize it. And so this is exactly the second mechanism of defense that I'd like to bring to your attention, which is that even if a B cell escapes that reacts to self, almost always it's also going to need a T cell that reacts to self to come and activate it. And so you need both the B cell to escape the weeding out in the bone marrow and the T cell to escape the weeding out in the thymus for you to get an active B cell that's now going to start putting out antibodies that react to self. And by the way, this cellular kiss here is usually going on in the lymph node. Now, looking at this whole process, you might have a few complaints, and I encourage you to think about how it might go wrong. Well, one way you might think it could go wrong is, what if bacteria got into the bone marrow? And certainly that's very, very possible, because when you get infections, the thing that's infecting you can move around your body. So if this bacterium gets into your bone marrow, does that mean that now this B cell is going to bind to it and therefore is going to be killed? Because at this stage, whenever the B cells bind to something, they're killed. And the answer is yes, this is exactly what happens. But the reason why it's not too much of a problem is that even if you have this bacterium here in the bone marrow for a week or two, or maybe a month, after that, once this bacterium goes away or is killed, then it won't be there anymore, and you can start producing these B cells that react to that bacterium again. And hopefully you already had a bunch of these B cells that could react to this bacterium that you had made previously and that were already out in the lymph nodes. And so those guys will be there to fight the infection, while maybe the infection might be in your bone marrow, preventing you from making more of those B cells to kill it. So you already have some of those B cells out there in the lymph nodes, and they can proliferate out there and sort of lead the battle from there. Now, even though your body has these mechanisms to keep your immune system from reacting to yourself, it still happens sometimes. The process still goes wrong sometimes, and the result is autoimmune disease. It's called autoimmune because you're immune to yourself. And so your immune system basically starts attacking your own body, and some pretty terrible diseases can result. And so to kind of bring it to life for you, I'd like to tell you about one example of this. So this is a muscle fiber, and the way your muscle fibers are activated, because you don't want to be flexing all of your muscles all the time, the way that they're activated is that they have a little receptor, which I'll draw here. And this receptor is ready to receive little molecules from a neuron, part of a nerve, receives little molecules from this neuron that activate this receptor and therefore activate the muscle fiber. So if you want to tighten this muscle fiber, you just need to send a signal down the neuron and it'll release the little molecules which will activate the muscle fiber. But in one example of autoimmune disease, you get antibodies against this receptor here on the muscle. And so they bind to it, and that either stops it from functioning, makes it impossible for it to react to the neuron's signals, or causes that receptor to be destroyed. And these are two mechanisms that have been seen. And so in this autoimmune disease, what do you think will happen? Well, what happens is that molecules from a neuron, part of a nerve, receives little molecules from this neuron that activate this receptor and therefore activate the muscle fiber. And so if you want to tighten this muscle fiber, you just need to send a signal down the neuron and it'll release these little molecules which will activate the muscle fiber. But in one example of autoimmune disease, you get antibodies against this receptor here on the muscle. And so they bind to it, and that either stops it from functioning, makes it impossible for it to react to the neuron's signals, or causes that receptor to be destroyed. And those are two mechanisms that have been seen. And so in this autoimmune disease, what do you think will happen? Well, what happens is that your body can no longer activate muscle fibers as easily. And the disease is called myasthenia gravis. And the etymology of that is my sort of means muscle, and asthenia means weakness. So muscle weakness, and gravis just means it's serious, because it gets serious over time. If you can't activate the muscle fibers in your body, you slowly become paralyzed. So you don't need to remember this exact mechanism. That's not really important, but I just wanted to give an example of one kind of autoimmune disease and how it works. So we've talked a lot about different cells in the immune system and what they do and how they do it. But one thing we haven't talked much about is where they do it.
Pause. Give me phone, Obama phone.
So in this video, I'd like to talk a little bit about exactly where the different cells are acting and how they move around. So let's start with neutrophils. And neutrophils circulate in the blood. So we're going to draw a little cartoon of blood circulation here. So blood circulates in a closed loop. I'm leaving out the heart, I'm leaving out the lungs, all that. Let's just say blood is circulating round and round like this. So neutrophils circulate in the blood. So let's draw a couple of neutrophils there. They're actually the most common immune cell. These are neutrophils, and in case you don't remember how to spell that, let's write it here, neutrophil. And neutrophils will circulate round and round the blood, but that's not actually where they do their work. They do their work only when there's infection, and that infection usually occurs out in the tissues. So let's say we've got an infection out here, a little bacterial infection. Now the neutrophils are needed, but they're needed out here. So the question is, how are they going to get there? And the answer is, well, they're going to have to squeeze out through the cells of the blood vessels to get to this tissue. But the follow-up question to that is, how do they know to squeeze out here and not to squeeze out here, for example? And so it turns out that you have these cells out here called macrophages, which I hope you've heard of by now. Macrophage, let me write that out for you as well. And those macrophages are phagocytes. You have the word phage in their name, so you could probably guess that. They're phagocytes, so they're going to gobble up some of these bacteria. We draw a couple of them in there. And they're sort of going to go into action because they'll notice that there are bacteria there. And when they're in action, they're going to release some chemical signals. We don't need to really go into conversation about what those are, but they're going to release some signals that will then tell the endothelial cells, here, I'm going to draw a couple of them, that are going to alert the endothelial cells lining the blood vessels that there is an infection here in this tissue. And as a result, these endothelial cells are now going to express these little proteins here. This is very detailed, but just to give you a sense of sort of the flow of information here. They're going to express these little proteins here that are going to stick to those neutrophils. Those neutrophils are going to be coming by, and they're going to get stuck on these little proteins that attract them. They'll be stuck there, and then because they're stuck there, they'll have the opportunity to squeeze through between these cells. And obviously other proteins and stuff are involved there, but that's a whole lot of detail that we don't really need right now. The neutrophils are going to get out here, and do you recall what the neutrophils are going to do? Well, the neutrophils are also phagocytes, and so they're going to gobble up a bunch of these bacteria. They're going to gobble them up, chew them up, digest them, whatnot. And then once they've done that, they're just going to die. That is their job, is just to eat up some of the bacteria to get rid of it, because you want to get rid of it. So they're just going to die, and it actually happens pretty quickly. Their usual lifespan is only 5 days, and once they die, they become pus. So a little later, that's what you see when you see pus. But really, the important thing in this whole diagram is this process by which the neutrophils squeeze out between these endothelial cells. Because usually there's only a very, very small hole. There are only very small holes between endothelial cells, and certainly not big enough for a cell. So it's an active process by which the neutrophil kind of pries apart two endothelial cells to squeeze through, and afterwards, the endothelial cells come back together again to seal off the blood vessel. And it's worth noting that immune cells only ever go in this direction. They only cross from the blood into the tissue, and it's actually impossible for them to go from the tissue back into the blood. And so that raises a question, because in previous videos, we said that the macrophages start here, they eat up some of these bacteria, and then they want to go present these bacteria to B and T cells. They want to show these bacteria to actually really, especially T cells, so that the T cells that are capable of fighting this infection can come fight it. But the B and T cells that are going to fight this are not located out here in the tissue. And so the macrophage needs to go to them. And so then you might have the question, well, does the macrophage come from this tissue into the blood and go find the B and T cells? And the answer, as we've said, is no, because it's impossible for cells to go from the tissue back into circulation. And when I say impossible, I'm sure there are some exceptions, but for the most part, it doesn't happen. And so the answer to this conundrum is that these macrophages need to use a different mode of transport to get to the B and T cells. And I'm drawing that here, and this is a lymphatic vessel. And if you're totally unfamiliar with this, perhaps afterwards you should go watch some lymphatic system videos. But lymphatic vessels have little holes in them that actually make it fairly easy for cells and proteins and other things to get in. So the macrophage is going to get into the lymphatic vessel, and that's going to allow it to travel to the nearest lymph node. And so here we've got a lymph node, and it has some T cells right there, and some B cells as well. And it's there that the macrophage can do its very important action, which we'll mention here because it's important. We'll draw a little star there. The star is antigen presentation. Antigen presentation, and there are other videos on that, so we won't really go into that here, but it's, I think, useful to know that that's where it happens. And as a result of antigen presentation, hopefully we have some B and T cells that are specialized to fight this bacterial invader. Hopefully we found them, and now they can go join the fight. So what's the obvious question? It's, well, how are they going to join the fight? How are these guys going to get there? They do not travel backwards through the lymphatic vessel because lymphatic flow is one directional. It flows this way. But luckily, lymphatic vessels all end up draining into the blood. And this is our cartoon, but if this weren't a cartoon, this end lymphatic vessel where the lymph fluid is going back into the blood would be called the thoracic duct. I'm just mentioning that for orientation. So luckily, all the lymph in the body, the vast majority actually goes directly through the thoracic duct, which is this, this fat, final lymphatic vessel that drains into one of the big veins of your body. But really, all you need to know is all lymph eventually drains into blood. And so that's exactly the method that these T cells can use to get back into the blood. And now, once they're in the blood, what are they going to do? Well, they're going to do essentially the same thing as these neutrophils. And maybe the chemicals, the proteins are slightly different, but essentially it's the same process. They'll come here, they'll realize that this is where they need to go because of certain chemical signals, and then they'll squeeze through between the endothelial cells and get here. And I'll try to draw a T cell out here, even though there's not much space left, so that you can see the points. And note that we also have B cells. And you might be wondering, are the B cells going to go out and do the same thing? And the answer is that some of them might, but B cells don't really need to be out here, because what B cells are going to do is release antibodies. Here are some antibodies. You're going to release antibodies which can actually just float around the body themselves. And they'll draw a couple around the blood, and some of them will get out here to the tissues where they need to do their work. So B cells, for the most part, can kind of chill out in the lymph node here, which I hope I mentioned that this is called the lymph node. If not, let me write that. They're hanging out in the lymph node there, pumping out these little antibodies who can then go travel and join the fight themselves. All over our body, we have blood vessels. And in those blood vessels, we've talked about how we have lots of different kinds of blood cells. So we have some red blood cells that I'm drawing here. But if you watched our immune system videos, you also know that we have some T cells and some B cells and some macrophages and some platelets. And all in all, actually, there's pretty much 10 different kinds of blood cells that we have in our blood at all times. In this video, I'd like to talk a little bit about where they're made, which you might already know, and also from what precursor cells they're made. Because cells don't just come out of nowhere, they come from precursor cells that divide and produce new ones. So do you know where all these cells come from, where they're made? I'm going to draw the answer. Cartoonish as always, and not to scale with this blood vessel. So here's a bone, and the answer is that all these cells come from the bone marrow, which is inside the bone here. Now, when I first heard this, I thought to myself, how is it possible that blood cells come from inside the bone, get through the surface of the bone, and get into blood vessels? Well, you might be surprised to know that all these bones are actually perfused with blood vessels themselves. They're just very small and difficult to see. So it's actually quite easy for cells to hop into these blood vessels and go into blood vessels of the body. But now let's talk about those precursor cells. So it turns out, and this is something that we didn't know for a long time, but it turns out that there's one powerful stem cell in your bone marrow that can make all 10 kinds of blood cells. And that one cell, that very powerful cell, has a bit of a complicated name, which we'll write out here. It's called a pluripotent. Pluripotent, if you're good with your Latin, you know that this means, I guess, able to do a lot of things. That might be a poor translation, but anyway, pluripotent, hematopoietic, because hematopoiesis is the process of producing blood cells. So that's pluripotent hematopoietic stem cell, because stem cells, as you may know, are cells that can divide into multiple different kinds of cells. And now I'd like to ask you a question, which is, do you think that a macrophage, which if you watch the immunology videos, you know is a big phagocytic cell, so it likes to swallow invaders or debris, do you think that the macrophage is more closely related to a B cell, which as you know produces antibodies, or a red blood cell?
B cell.
You might be surprised by the answer. The answer is actually the macrophage is more closely related to the red blood cell, which is kind of weird because macrophages and B cells are both immune cells and red blood cells are not. This pluripotent hematopoietic stem cell gives rise to two main lineages, and I'll draw them here. The first is the myeloid lineage, and the other is the lymphoid lineage. So when this pluripotent cell here first divides, it can give rise to one of these or one of these. And these are also precursor cells to the 10 kinds of red blood cells that we actually find in our blood. So these are not the ones that are going to end up in our blood. These are guys who sit in the bone marrow and make the cells that end up in our blood. So now it turns out that this lymphoid progenitor cell can... And these are also that are going to end up in our blood.
10 kinds
these are
of blood cells. Oh.
Guys with lymphoid body. It makes an NK cell. It makes a B cell. And it can make a T cell. And this is 3 out of our 10 blood cells that we talked about. And so the other 7 are going to be from the myeloid lineage. And one of those is, as you could have guessed, the red blood cell. Another is the megakaryocyte, which you may not remember what it does. So I'll tell you in a moment, but try to remember. The megakaryocyte is actually what makes platelets. So the megakaryocyte buds off little pieces of its cytoplasm surrounded by membrane, and these are platelets. But the myeloid lineage also makes a bunch of immune cells in addition to the red blood cells and the platelets. And you may not have heard of these, so I'll just list them here. One of them is the neutrophil. That one you're most likely to have heard of. And there's 2 other cells which are very similar to the neutrophil. One is called the basophil, and one is called the eosinophil. And don't feel too bad about not having heard about these because they're actually pretty rare in your blood. They are there, but there aren't many of them. And then there's also a monocyte, which is fairly similar to these 3 above. And the monocyte is actually what becomes a macrophage later on. You're probably familiar with macrophage. Monocyte is just a slightly less differentiated version of a macrophage. And finally, there's also mast cells. And mast cells are the ones that... So these are our 7 other cells.
What?
These 3 above. And the monocyte is actually what becomes a macrophage later on. You're probably familiar with macrophage. Monocyte is just a slightly less differentiated version of a macrophage. And finally, there's also mast cells. And mast cells are the ones that... So these are our 7 other...
Mast cells. Mast cells are the ones that ... Oh my goodness. It's funny. Did nobody catch that? Okay.
Other cells.
I don't know why I find that so funny.
And as you can see, what we said in the very beginning is in fact true. The red blood cell is more closely related to the monocyte, the macrophage, than is a B cell or a T cell or something like that. And now I'm adding an addendum to this video to mention one other kind of cell that we've talked about in the past videos, but that I didn't put on this chart. And that kind of cell is the dendritic cell. It's one of the antigen-presenting cells that we talked about. And I want to ask you, do you think the dendritic cell comes from the myeloid lineage or the lymphoid lineage?
Both, right?
And it's actually a trick question because the answer is it comes from both. So we have some dendritic cells that I'm drawing here, which come from the myeloid lineage, and some, which I'm drawing right next to it, which come from the lymphoid lineage. And it turns out that the ones that come from the myeloid lineage are created from monocytes, whereas the ones which come from the lymphoid lineage are not really descendants of B or T cells. They come from some lymphoid precursor. So dendritic cells are kind of a weird exception. They can be made from either line. And I wanted to add yet another piece of information, hopefully not overloading this too much, just to remind you once again that monocytes, in addition to becoming dendritic cells, can also become macrophages. And macrophages we are certainly familiar with, at least we've talked about them in the other videos. So here's a macrophage. And macrophages, if you remember, are kind of like sentinels. They sit out in the tissues and watch for invaders. And dendritic cells also behave like that. So what about this monocyte? Does he do the same thing? Well, actually, no. It turns out the monocyte is kind of like a circulating version of macrophages or dendritic cells. So what do I mean by circulating? I mean that it's actually in the blood. So monocytes move around in the blood, and then when they go into the tissues, they settle down and become sentinels. That's when they are turned into either macrophages or dendritic cells.
Oh, there you go. Somebody caught it. He did not tell us. Yes, he caught it. That's fine. Okay. Great. Great. All right, maybe let's take another 10 minutes before we start the questions. And actually, before we start the questions, I do want to go over 238. Let's go over 238. Tomorrow, the sun will come out tomorrow. 238 to 242. 238, 242. Hi. Yeah, so let's take a break. We'll read this and then we'll do the questions. And if it's a little bit too low, then I think we'll watch some 100K. I still do want to watch a couple 100K videos. Because he's just that good. He's just that good. But we'll see. We'll have to see. It depends. He might just go into it a bit too much depth, so I don't know if it might be too much. But I don't think so. I mean, I'd rather know more than know too little. Check it out. Do you have any comments? Hey, Nadine. Yeah, good luck. Good luck. Let us know. I guess, yeah, let me know. Let me know how it goes for you tomorrow. I guess rest and prepare well. Right, yeah, just take it easy, rest, prepare well. Prepare well. as well. Sadness, fluid emotion. Anyways, go back to 10. Yeah, okay, let's take a break. Like I said, let's take a break. It's getting hot again. I shouldn't be taking too many breaks. I'm only human. Okay, yeah. I'm not here. Two 38. 238. Set height. Set height. Yeah. Um, okay. Yep. Go for it. Excuse me. MHC2, MHC1. Oh, B cells be wildin'. Humoral, be extra humorous, be extra. Oh, humoral, be extra humorous. MH2 is so fun. Big Mac Daddy. These are not Why does that hurt? Cigarette explanation. I guess that's it, huh? Okay. Not that much on that one. Big Mac Daddy. Big Mac Daddy. Big Mac Daddy. Big Mac Daddy. Monocytes, leukocytes, white blood cell. Monocyte, I think we'll go to macro. Neutrophil. I think it's macrophage. Innate. B cells, macrophages, dendritic cells. Well, I mean, this one is in both. Well, no, that's T cell. The bone marrow. The bone marrow. I think it's antibodies. No, it's gonna be the B cells, I think. B cells first because then they ingest and digest, and then antigen. What was it? Antigen showing cells or something? Anyways, I think it's Wait, what? Adaptive. Wasn't it in the thymus? It wasn't in the thymus. That was the bone marrow. Why is it bursa of fabricius? B cells own antigens. I don't think it's in the thymus. In the thymus? Oh, wait. It's antigens. Wait, what? What the heck? The video said, I could swear the video said, I could have sworn the video said it was in the bone marrow. No?
In fact, view those receptors here. And let's say that recognizes this insulin protein. I'm going to take that protein and going to picture that you weed out all the B cells that will be things to non-self things and not to self things. So the processes we're going to talk about are equally true, maybe even in some cases more so true for T cells than B cells. So let's go to the bone marrow to figure out how this process works. I'm going to the bone marrow to show a small amount, but it'll be there. But this recognizes self. If it sees that embedded on it, not, and maybe you have every single protein here of two pools or different other cells around, and this B cell has matured and become different. So the rules are different, and he's not going to die. This weeding out of B cells that react to self proteins is sort of the first of two mechanisms that I'd like to talk about that the body uses to not react to self. And actually, the same exact thing happens for T cells, except it doesn't happen in the bone marrow. It happens in the thymus because that's where T cells mature. So in the thymus, we really the identical process where T cells differentiate, and each one has a unique receptor, and the ones that react to self in the thymus too strongly are killed. But it's not a foolproof method, or else you wouldn't need step 2. Body will interact, and they're going to antibodies that react to self. The problem is that even if you have this bacterium here in the bone marrow, today, and so but what happens is that
I forgot about that one. Okay. Wait, what are we talking about here? Yes? No. No. The overtolerance? Oh yeah, we did. That's what we just saw, I think. Where's clonal selection? Yes, I think. No. Not specific. No, that would be bad. No. Are places where we wouldn't circulate back to the blood. I would think we wouldn't really see it in the appendix, because why would it be there? Okay, that's not bad. That's not bad. Hold on, let me just check this out real quick. Yeah. 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 Okay. think so. That's a good question. I think it's, I mean, for sure C, I think. It's for sure true. And this is true for both. I think to fight, probably the innate, right? No, probably not the innate. So it's kind of backwards. So I think it's false. Does it make sense? TLRs. Oh, versus whatever versus the receptor is. Oh no, I'm just gonna whatever. Something. Okay, TLR. Yeah. I can't remember. Yeah. So, in the cell wall, probably not inside of it. I don't think so. Microphages. Cell adhesion. Why? What the? Am I reading this wrong? Oh, actually, maybe B actually makes sense. Yeah. Okay. Actually, I don't think it would be antigen, would it? I thought it would be protein. Yeah. Yeah, it's specific to the passage. Specific. Neutrophils. Okay. That's a good question. Um, oh man. Would they not expect misfolded? So it's misfolded T cell receptor. That means it's not going to bind. It's not going to bind. That means it's not going to activate a response. I mean, just it's not going to T helper to B cell CD4 plus to activate the B cell, right? To then go to produce more antibodies, or turn to a plasma, or turn to a memory cell, or go into CD8 plus enter into cytotoxic T cell to destroy the mutagen. Yeah, I guess that's right. So we would not expect. So this is stuff we would expect. This is all normal. I don't think we'd expect the immune system to attack the host. Unless we know for sure that this mutagen was part of the mouse self. We would expect B cells to not produce antibodies because it is misfolded. So this is expected. Infected cells are not apoptized. This is expected. Innate immune system does not activate. I guess this is expected, right? Is that expected? I thought it would be the adaptive. I don't know. Unless we know the mutagen itself, then the innate immune system. Oh, well, if it's required to activate the adaptive, then that's expected. So it's a... What? Would they not expect to see? Would they not expect to see? Would they not expect to see? What? Pretty sure we ain't. What do you mean not quite? I'm missing something. I guess the DNA immune system does not activate. Wait, why though? They would not expect to see, as a result of the misfolded TCR, that the innate immune system does not activate. Wait, that the innate immune system does not activate. Oh, we would expect. We would expect the immune system to attack the host. We would expect the immune system to attack the host. What? Um, immune responses of first line. Oh, the immune system attacks the host. Oh, did I miss something? The immune system attacks the host. Okay. Um, multipotent, pluripotent. I think pluripotent. Ooh. Multipotent? Multipotent. I thought it was pluripotent. Pluripotent. Multipotent? Multipotent? Oh, that's the all-star cards. Okay, pointing out. Okay. What? Something antigens to T cells. I thought that uses MHC1. Oh, that's B cells. Oh, never mind. That's interesting. Um, oh, B cells. Okay. Sure. Wait, let me think. Least expect. Neutrophils? No. Cytotoxic disease. Oh. Okay. Right. Yeah. That makes sense. These are kicking my butt. Um, think that's true. I don't think neutrophils. I still think circulating blood. What? Oh, okay, they do. Yeah, so. It's not only into the tissues. Once it's not like in interstitial fluids, which means they do actually go into the bloodstream. Okay, never mind. Negligent. The fact the second one had a faster increase than the third brother. Let me see. Dysfunction of which of the following cells? Dysfunction of which of the following cells could explain the reaction? First brother. The first brother? See, so I think it's either neither, or I think it could be B cells. Right? I could be wrong. He's got no globulin, so he's not producing as much antibodies. He's not producing as much antibodies. That means that the B cells are not being activated. that means that there's that there's some sort of issue with digestion of the antigen presenting cells. I don't think it's between the B or T cells. Okay. Never mind. Okay, so I think it would be between. Then it would just be between the B cells not having those specific. Or actually, between the B cells not being activated. Okay, both, right? Because B cells need to be activated by T helper cells. Yeah. 3 out of 5. Dang it. Ah! $30, $40, $60. 30, 40, 60, 80. So this might be easier too. And it looks like Xan 2 is more effective, right? Xan 1 is not, because they're dying at day 6 post-infection. So Xan 2 is more effective. Okay. How's the one thing under control? I'm not sure much different than oh, jeez. Active immunity. Emphasize very challenged. The individual is giving an attenuated form of the pathogen. Such as vaccines. Okay. Lifelong immunity. I don't think so. It's more like sleep for the West. So, so plasma cells or memory cells. Plasma cells give out a bunch of antibodies. Memory cells just hold it. It's in the blood. I think um, actually maybe just plasma then, right? effect on the cells. Okay. Yeah, so that means it's in the blood. So we do have this occurring. We don't have memory occurring. Oh, let's go. Ah. Oh. Okay. Okay. This is okay. Jeez. Okay. Yeah, the plasma rate is increasing. It looks like ZMAP2 will have the highest plasma selectivity. ZMAP1 does not. Because they're dying. What? Wait, what? They're dying. Your X is dying, right? Percent survival. What does this mean? Some people are dying? What does the X mean here, man? I mean, Jesus, man. Seriously? Maybe it's impossible. It's impossible, or it's a control now. Okay. It's impossible. Can you stop? Seriously? Why'd you have to come to my room, man? Oh, actually, that is true. That's a good point. Yeah. Oh, that's true. It's definitely pro-inflammatory. So much that it. It's not anti-inflammatory. I don't think it's anti-inflammatory at all. And We have too little of it. We need to increase production. I think that'd be positive. Wow, let's go. Still, that's not that good. It's only 3 out of 5. Where's this fly? What the heck? Seriously? You're gonna fly in the room? I bet you when I sit down, it's gonna come to me. Come here, fly. Man, I gotta freaking fly in the room. Oh, I see it. Wait, nobody move. Nobody move. Ah, I missed it. Come back, where'd you go? Find no word and then it's gonna bother me again. Seriously? Whatever. I'll be ready. Nice one. It's just about the same. I guess. What's next? Well, what's next is Anki, probably. Yeah. Oh, it's right there. Where'd it go? I'm pretty sure I didn't get it. I don't think I got it. Where did it go? It's hiding. Stop hiding from me. Just land. Ah, yes. Okay. I can't tell. Did I get it? I assume I got it. Imagine. You get a second there. This is. That would be funny. That would be something. I feel like I got it. I think I got it. Oh, what's that? I can't see. All right, I'll be back. All right, let's. I think I got the fly. I don't know where it's at, but I think I got it. Also, I think. Yes, I do. Not gonna mess with that. Okay. Yeah. Decks. Just go through the deck. It doesn't matter. What? Was it like this last time? I don't know if it was. Could have been. Yeah. Well, it's a little weird on your guys' end. There we go. What happened to the fan? We're literally getting cooked. Turn it up all the way, dude. Digesting again, That's fine. Add a little bit extra in here. Is it a whole new system? Seems like it. Oh my God, look at that. Oh, I didn't know. Yeah. Yep. Yeah, maybe let me just draw it there, I think. This for now. We will need it, but not right now. Switch away, buddy. I don't know what happened to the slide, but I think we got it. Yeah, now, wow. We're doing great. We did great. I think, well, somewhat. Anyways, let's go ahead and reset now. Yeah. Yeah, let's go ahead and reset. It's freaking hot, man. What's the tempo? (82, 25% humidity. I don't know, I think it's gotta be wrong. I think it's always wrong. Like right now. Who's it at right now? (82). I don't think it's gotta be more. I think it's gotta be more in my room. I don't know why, but I think it's always just way more in my room. But anyways, yeah. So let's reset. Yeah, let's reset. Let's get a quick workout in. I still want to watch those videos, but it's gonna take some time. Maybe tomorrow morning. It's gonna be 4 hours plus 2, so I need 6 hours. Start at 6. We'll see. We'll see. Everything's possible. All right, I'll be back. Hold on. Oh, actually, this works. All right. Let's go ahead and look at it. Yeah, sorry, the bookshelf's a mess. Here's what it is. Oh, I know. Let me see. Just over here or something. There we go. I need some eye drops on my eyeballs. Those are kind of different. So let's put them up there. Yeah, so what I want to do before we start the physics is close out any of the lingering biology gaps, which the gaps, I think some of it was caused by it's just it wasn't really in the Khan Academy videos. There's some that take you to another site that are on the Yankee cards that I don't know if they're going to be on there or not. I don't think so. But I don't know. I don't really know for sure, man. Yeah. Anyways, let's get something going here. Get up. I swear, I hear police sirens almost like every day. Yeah, that's good. It means we're doing a job, right? That's good. I don't know why. I think I want like what do I need? I don't know. Where's my glasses? Oh. About this bandana thing? I don't really know how to wear it. But this is fine. Something like this is okay. This will do. Yeah, this is okay. So kind of a little bit like more leg focused. Because I'm still recovering from I'm still recovering from a bit of time, so oh yeah, actually oh I shouldn't. Let's see. Oh, my God, it's a bit now. Guess I'm looking tired. Whatever. You know. I don't really feel anything when I get to the top. The tension is at the bottom, so that's why I want to stay. You know? Let's go. All right, I'm gonna take a quick cold shower. Yeah, it's getting way too hot. It's gotta be at least above 100 right now. There's no way. Current temperature. It's gotta be like 90 something with this humidity. No way. I don't believe it. I mean, it takes a lot for me to sweat even when I'm exercising. I'm freaking, like, yeah, there's no way. Let's see, at what temperature does the iPhone get a heat warning? What temperature does the iPhone get a heat warning? Yeah, 95. Yeah, see, there's this way hotter in my room. All right, whatever. Anyways, let's go ahead and let's go ahead and take that shower, right? Oh, that was a killer leg workout. And I shall be back. I will leave you guys on the Yankee. Hey, sister, you still make me wonder. Oh, and the sky comes falling for you. You're sleeping in the you. Yeah. All right. Well, let me fix that. Oh. Mm. Yep. 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 You know, if I can pay with that. Let's see. It depends if GameStop pops. I hope it does. This time, I'm pulling out. So good. So good. Too good. bodies. Yeah, whatever. All right, it's concerned. My feet. Something like that. Protection areas. I think you ready to tear a little bit. A little more. Yeah, that's good. That's hot. No. Save the water for later. Stop. Antibodies. High-low microns. Okay, let's take a look at it now. Okay. That's the lumen. And the field itself. Oh, that's so good. That's so good. That's some high quality H2O. Anybody here ever watch Forrest Gump? Is it that? No, not Waterboy. Waterboy, not Forrest Gump. Oh wow. Cooperative? No. Yeah, cooperative. Let's go. Selectins. Okay. Neurotransmitters. Antigens. Specific antigens. Selectins. Got it. IC2 antigens. No, ADP2 carbohydrates. Terpenes. Anybody? By a suppressor, repressor, substrate. Oh, Smell, speech recognition. I think hearing sound, right? Hearing sound. Hearing sound, speech recognition. Yeah, that right? Oh, I forgot memory and emotion. Again. By the product that is formed beyond subsequent pathway. The product that is formed way later in the pathway. High levels. In the same pathway. Oh, well then. Antibodies? Competes. Mixed with each other? Oh, okay, yeah. Synthetic. High levels of product that is formed later down in the same sequence. Or the same pathway, yes. Finding timers. That's a good question. Visual bingo. I don't remember. Oh, that's right. What the heck? Why is it red? I don't want it to be red, buddy. Same thing with you, huh? Smooth movements are fine. Smooth movements and boisterous loading. binds at the ES complex. Combine allosterically. Bmax goes down, Km goes down. Bmax goes down, Km goes down. What else? Hamburger plot shifts up. Morning. Sensory processing. Yeah. Non-competitive. Bind allosterically. That goes down. Okay, no change. Mine at the E or ES or ES site. You can also get positive or negative binding effectors, which will increase or decrease binding. And then both points start at the x-axis. With one line having a higher slope. Okay, specific answers. 4F. Fighting, fleeing, feeding, fornicating. Integrates in the endocrine system to control the pituitary gland and hormone. Hypothalamus. Forebrain. Competitive inhibition. Selectance. I've noted once, and is oh man. Let me think about this. Let's do it. Oh, excuse me. It's a lot of water, basically. Mm-hmm. Proofreads and exercises. Audio sensory functions. Very cortex. Maybe audio sensory function. Anyway, if we have speech recognition, sound, memory, emotion. Yeah. Hearing. It's auditory area. Not the better vision. Active sentence competes with the subject. Oh yeah. Basal ganglia. The basal ganglia. Homeostasis. Isomerase. Also glucose isomerase. Emitted by high levels of the final product within the same feedback inhibition pathway. inverse plot. And the slope is km over v max. A double reciprocal. That's right, double reciprocal. Somatic and automatic nervous system. Hyperbolic. Suicide. Suicide is different. Double link membrane chain. 2. Smooth muscle and osseous glia. Activators. That's the EF site only. Suicide inhibition is a form of enzyme inhibition that occurs while it's an irreversible inhibition and is a form when a separate covalent bond covalently bonds to the active site. Simple transport, passive transport. Oh. Dang it. Whatever. Contains so strange. Vessels. Yeah, okay. So emulsified by bile salts to form micelles. Micelles? How do you call that? Micelles or micelles? So you think micelles. Oh my goodness. Oh, my cells. Okay, my cells. And I send them out to chylomicrons for releasing the lymphatic system. Oh, it's epithelial cells, not endothelial. Epithelial cells. I was right. What the heck? Dang it. Oh. An outside regulator that is also the substrate. Neurons. Dendronucleosoma, myelin sheath goes around here, axon hillock, active potential that way, and all the other stuff. Blah blah blah. Okay. Parasympathetic, allosteric inhibitor that is also the substrate. Oh, heterotropic. And it's the allosteric regulator. On the ligand strain? Allosteric regulator that is also the substrate. Okay. Carbohydrates, lipids. Glycosylation. Is glucose? Wrong carbohydrate. Kotler neat sugar. 309 asparagine. And serine. And asparagine is N-lane. Serine and 309 are Olink. Allosteric regulator that is different than the substrate. Temporal lobe. Central nervous system. That acts as a binding molecule? What are we talking about? Okay, yeah. Sure. Oh man. DNA molecule. Eukaryotes have just kind of these long, whirly things, but they also have plasmids. If you think about it, they kind of are still circular. Centrosimonic circle. In a P, in an A site. GC content. Proteins on the surface of an antigen, or proteins on the surface of a pathogen, or I don't know, or virus maybe, or toxin. Well, that's fine. Nerves and fibrous tissue not found in the CNS. And you have essentially just nerves, cranial nerves, uh, cranial nerves, facial nerves, spinal nerves, different nerves that will, send signals back to the CNS. Sensory neurons and motor neurons. CNS only contains breathing neurons, interneurons. Cortisol. Why is it always such thing? Glutamate. That's a good question. On the surface of other cells and using the immune system. Mismatch repairs. Adrenal cortex. The X-intercept and the Y-intercept. Nucleic acids. Steroid precursors. The surface of cell molecules that are found in the immune system. Yeah, basically. Facilitated diffusion. Proteins on the surface of a pathogen or toxin. Found on the surface of cells that are most commonly found in the immune system. What was that? Anki? Yeah, that was Anki. Its functionality. Codes for another stop codon. facial bones, swan cells, melanocytes, adrenal medulla. Non-proteins. Genetic code that will code for genetic code that will be translated into proteins. Differentiation, interactions, differentiation. Estimated potential is negative 55 millivolts. Completely wrong. It is negative 70 millivolts. There we go. Todd is uptight, indicating he has no oxygen bound to him and low affinity to the binding of oxygen. Relax. There's a couple glasses of oxygen attract him more. Yeah. There you go. Yeah. I think that might be 5, but let's go with good, I guess. Coenzymes are organic molecules found in vitamins and those within our diet. Yeah. Longitudinal axes, the longitudinal axes, and Sonic the Hedgehog. Oh, never mind. No, it's not Sonic. And eating and nervous system development. That was Sonic the Hedgehog. It's not Sonic the Hedgehog. Oh, it is Sonic the Hedgehog. What the heck? Oh no, the nodal cord. The nodal cord is for longitudinal axes, but the protein produced by the nodal cord is Sonic the Hedgehog. Sonic the Hedgehog. Sonic the Hedgehog. Yeah, I think for the spine. Did I get it right? No, I think I did. Controls plus channels open. Polarization. To a left shift are low temp, high pH, and low CO2. We're basically going from the lungs from the lungs to the tissues. Yes, sir. Oxygen dissociation curve. Yes, sir. Or yes, ma'am. DNA polymerase. You will place new nucleotides in the excised region. There we go. Sir. Saltatory conduction. Saltatory conduction. The mesoderm differentiates into man, what are you differentiating? No cord, maybe? No, somites. Somites. Homeostasis, aided enzymatic processing, and regulation of cellular respiration. Orchestrating. Orchestrating. Yeah, that's fine. Nice. Yeah, today was a good one for sure. I think we're nearly done. I think the rest we probably spend a little bit on that. Some practice on the things we've done, and yeah, and then just NVK videos, I think. Or just maybe set up for next week. We'll probably hit more cars and things like that. I think we'll be okay. We will be alright. We're gonna be alright. Okay, negative properties have less binding affinity. Recognize unchanged conformational structures. Lower binding affinity. Lower affinity is a process where DNA is synthesized from RNA. Post-transcriptional mod. 5' PAP. Poly tail. And splicing. I get those every time, dude. Differentiation, signaling interactions, lineage, signaling. What are we talking about? Oh, induction. Induction. Hemoglobin, heme group in hemoglobin. Oh, wait, what? What? Never mind. Metal ions. Yeah, that's fine too. Okay. Okay. Oh, I didn't even really read it. Whoa. A blastocyst. Axons. Jeez. Polyetail. Yeah. Positive or negative corporate derivative. Calcium 2 plus, magnesium 2 plus, Na plus, K plus maybe. Oh, maybe not K plus. Yeah, okay. Magnesium 2 plus, zinc 2 plus, calcium 2 plus. Magnesium, calcium, zinc. Magnesium, calcium, zinc. What did I say potassium? Magnesium, calcium, zinc. Autocrine, paracrine, autocrine, juxtacrine, paracrine, paracrine. Partial potential. Whose introns are sharing only exons. -5 millivolts. Positive cooperativity. The process of Planck removes non-protein co-ingredients. Splicing, zinc, calcium, magnesium. Tot is uptight, indicating no oxygens are bound to it, and a low oxygen binding affinity. Dane effect is shifted to the left. You go to dissociation curve, where the growing blood chemistry from the lungs under the conditions of low temp, low temp and low CO2 and a higher pH. And mitosis results in 4 daughter cells, each having half the number of the chromosomes as the parents. H2HB is bound to oxygen. If Keck is equal to Q, the delta G in the equation, delta J is equal to delta G prime plus R T of natural log Q will be 0, signifying that equilibrium has been reached. Coenzyme or cofactor. Germ cell. And development of the nervous system. Yeah. Activators are essential component. Activators are essential. Or changing of the codons of the amino acid. Failure of DNA repair mechanisms. RNA reads. Somites. Activators or helpers to the active site. Helpers the composition of the active site. Oh. Activators or components. Activators or components of the active site. Quaternary structure. What the heck? What are you talking about here, mate? What the heck does that mean? Okay, quaternary rhythm, 2 alpha and 2 beta subunits. Oh, that's an old one. The peptide bond is formed between the amino group nitrogen, which acts as a nucleophile, and the wait. Yeah, the amino group nitrogen, which acts as a nucleophile, and the carboxyl. No, the amine group of the amino that you're trying to add, which acts as nucleophile, and the carbonyl carbon of the amino you already had, which acts as the electrophile. Is that right? It's technically C. I mean, I'm sorry. It's technically well, C double O to the N, right? But the C is the one you already had. The N is the one that's being added. Electrophile, electron acceptors, nucleophile, electron donor. Activators or active site components or enzyme components. Induction. 2 alpha subunits and 2 beta subunits. Less binding affinity. Move of the introns. Cannot fire immediately. Cannot fire another action potential immediately, allowing the signal to move in one direction. Immediately. DNA mutation occurs when there's a change in the nucleotide of the amino acid codon sequence. Nucleotide codon sequence. The DNA. In that DNA, tries the protein's affinity to want to bind more to that specific substrate. That's the same thing, I guess. Michaelis-Menten equation. Okay, Michaelis-Menten kinetics. Myelination. Equivalent to the bonds, the energy stored in the bonds, or equivalent to total heat energy within the system. Also, the equation is delta H is equal to E internal energy plus P times V. Energy stored in the bonds. The energy stored in the bonds. The energy stored in the bonds. James Bond. 007. Negative side with negatively charged substrate. Into the positive amino acid like positively charged amino acid like lysine, histidine. Lysine, arginine, histidine. Lysine, arginine, histidine. Factors contribute to a shift in the O2 dissociation curve. A right shift, we have high temp, low pH, and high CO2, and also high 2,3 BPG, which is the same thing as 2,3 DPG. But it's not there. I should probably just add it, huh? Do-do-do-do-do-do-do. Does that work? There we go. Positive cooperativity. The relaxed state, HB, is bound to O2. Yeah, I had 29. It's crazy. After repolarization, continued loss of K2 leads to lowering the voltage. Leads to repolarization, which leads to hyperpolarization. Blank is a non-protein entity crucial for a protein's function. A cofactor. Did you know that? It's a cofactor. Color difference. Did you know about the color difference between oxygenated and deoxygenated? So there are the two configurations of O2. Oxygenated blood appears, and it is relaxed, it appears bright red. And when it is deoxygenated blood, it appears darker. It's flowing in my veins. My veins. 2K plus go in. Right shift indicates high temp, low pH, increased CO2, and increased 2,3 dPg or 2,3 dPg. Oh wait, what am I talking about? Well, yeah, that's true too. Yes, that is true too. The key memory numbers to remember are some memory potential is peak count is around plus 40 millivolts. Is to send signals out. Signals out for communication between different neurons, right? Between different neurons, to different cells. Oh, and then send that signal back as well. Neuromuscular junction, so our neurons is sensory environment really. I think it's fine. I understand my understanding of your understanding of my understanding. That's from the new show, the Netflix show. It's the one with the little kid. Anyways, process splicing removes non-protein coordinates by ribosomes. A decrease in binding effect, MHB binding effect. It's gotta drop them off. It's gotta get dropped off. Between proteins and cofactors is hemoglobin. Hemoglobin's heme group. Hemoglobin's heme group. Okay? Hemoglobin's heme group. Exponential is more of a exponential is more of an event. RNA firing Silicon between across neural motion junction and relay that information back to the CNS. Yes, sir. Is to bind, transport, and drop off. Okay. Look at all that. Look at all that. Longitudinal axes. Left shift indicates an increase in HP affinity of filter. I understand. Refractory period. Negative 90 MV. Negative 55 MV. Hemoglobin's heme group, which binds to O2 through the F2 Fe2 plus contained within it. The hemoglobin's coordinate structure, which has 2 alpha subunits and 2 beta subunits, corresponding to whether they're bound to O2 and the binding affinity for O2 as well. This helps maintain threshold potential. No, resting member potential. Whoops, I'm not reading it. Then we get growth phase. A growth phase where we have development, production of membranes and organelle structures. Organelles. Prepare for cell division, which is M1, mitosis or meiosis. Hemoglobin is crafted from. What is it crafted from? What the heck? Oh, protein. Yeah. Okay. Darker red. And in the stomach. We also next as activators or social components. Their differentiation interactions. Development. Zinc, magnesium, calcium. Oh no, coenzymes. Oh no. What have you done? NAD plus and FAD. Those are the primers. DNA polymerase. Taut is uptight because it binds to O2, has a more higher binding affinity. Diploid state. Chromatin. Okay, suspend you. Breaks apart the DNA. Breaks apart the hydrogen bonds. And one's it, same thing. Alright, coding regions of genes and directly translated into proteins. Excellent. As a heme group, like a heme group, non-protein tightly bound molecule. By rapidly increasing the frequency of the action potential, essentially sending more action potentials. That gives a stronger signal. Describes the protein's binding affinity. Binding affinity to the substrate. And the diploid cells, somatic cells, mitosis. Higher binding affinity. Shift is to the left. Ascent strand. Oh, ascent strand. Yeah. As the mRNA. Is a protein, is a cofactor or molecule that is tightly bound. So non-protein that is tightly bound to the coenzyme. Or tightly bound. Tightly bound to the protein. Tightly bound to the molecule. Yeah. Okay. That's fine. High CO2, low pH, high temp. Increase. Decrease. Heme group. Hemoglobin heme group. Bind to the oxygen. The right shift indicates a decrease. Decrease. This helps maintain. This helps maintain. This helps maintain the unilateral flow of the action potential propagation. A left shift indicates an increase. HST configuration center left, corresponding to whether they're bound to O2 and their binding affinity. Cofactors and proteins. I don't know. Yeah, cofactors proteins. Darker red. Development. Coenzymes include NAD plus and FAD. It has a higher binding affinity. Life status relax because it has O2 bound to it and a higher binding affinity. Is a non-protein that is tightly bound to the enzyme. Or tightly bound to mitosis. Decrease in binding affinity. All right, guys, we're gonna take a break here. We're just a little bit. We're almost done though. Yeah, we're almost done. All right, I'm back. Let's wrap it up. I'm back, Hassan. Hassan. It's okay, buddy. I'm right here. Don't worry. It's okay. It's okay. I'm back. Let's go ahead and get started. Yeah. Okay, we got what do we got? Leucine, leucine Lu L, non-polar, hydrophobic, leucine Lu L. How many do we got? How many more do we have? Oh, this is the last one actually. Well, 4 is 4, so but that's a little, so that's okay. Yeah. Yes, sir. Brain and spinal cord. Plasma cell. B cells that do not become plasma cells become memory cells. We already have a blueprint for the antibodies that will kill it. I think, yeah. I have a little bit of a game plan for tomorrow, so we'll see. Yeah. Adaptive immunity. Oh, adaptive cell-mediated immunity. I'll go get another one. Acid. Key characteristics of the adaptive immune system. Big Mac Daddy. B cells, macrophages, and dendritic cells. Big Mac Daddy. Recessive alleles are seen to be masked by the presence of a dominant allele. Wait, let me think. Okay, recessive allele. Two recessive alleles phenotypically expressed. RNA poly binds to the region on the DNA called the Tata box. No, not Tata box. Promoter region. The Tata box. You got that Tata box? Tata box? Got that Tata box. Yeah, we're gonna close out a lot of stuff tomorrow. It'll be a great day tomorrow if everything can get done. I think it'll be like one of the greatest days if we can finish what we need to finish tomorrow. To the right, increasing temp. Shoot about boo. Yes, sir. We must have gone to fly, but I have no idea what happened to it. All I know is I was I was banging on that curtain, slapping the curtain with my shorts, and we killed the fly. So we killed the fly, and that's how it goes. Okay, decrease in temp. If we decrease the temp, we're gonna go towards the heat, and wait, hold on. If we increase the temp, then the shift the direction away from the heat towards the endothermic reaction. Let's think about that. So if we increase the temperature, we shift the direction of the reaction away from the heat towards an endothermic reaction, where endothermic is heat requiring. So if we decrease the temp, then we need to go towards the heat, going toward a when you go towards the heat. Wait. Away from the heat, towards a heat requiring. Oh, we decrease the temp toward the heat, toward exothermic reaction. It's interesting. Toward the heat in favor of an exothermic reaction. Decrease the temp. We need more heat. We need to release more heat. Oh, okay, yeah. So decrease the temp, we release more heat. Exothermic. Increase temp. Increase the temp. We need to go towards a heat requiring reaction to use up all the heat. Oh, okay, yeah, okay, I think so. Hold on, this fan's a bit too hot. B cells ingest a pathogen and present an antigen to a T cell, which activates the B cell to crank out a ton of antibodies called the plasma cell. It's very, very slow, it takes days. Post-transcriptional mod. Whoa, wait, what? Oops, never mind, I'm not reading it. Self cells. Pathogen versus self cells. Host cells. Some proteins require multiple polypeptide subunits. Coordinate structure. Pressure of that gas. The partial pressure. Taut state, uptight state. These are T helper cells marked by CD4 plus. CD4 plus. As the T helper cells marked by the CD4 plus activate the cytotoxic macrophages. See, that's where I wasn't sure. Yeah. That's true, actually. CD4 plus goes with MHC 2. Yes. Why do I get that mixed up in my head? Come on, brain, do better. Do better. Their goals was born of bacteria. Yeah. Yes. Hold on, though. Wait, what? Is that right? I don't think it's that right. I don't think that's right. I thought MHC2 go to CD4, and I thought that makes us activate the B cells to crank out the plasma cell and to crank out the. See, what the heck? I don't know. What? All right, am I messing something up? Am I mixing it up? I might be mixing it up, huh? Maybe we just watched the video now. The video's 26 minutes long. Oh, we will test operant conditioning. Seriously? Why? But why? We should I'll watch the video tomorrow. That's fine. Let's leave it here for now. Oh, I see. So essentially, T helper cells more support and help activate kind of the CT4 CD8 plus T cells and the B cells that can only bind to the antigen MHC2. I think we'll watch it. I'm sure we'll clear up tomorrow. We'll probably, anyways, we'll wake up tomorrow. I want to entertain a little bit of the IFD stuff. Cell to cell, adaptive immunity, cell to cell mediated immunity. Plasma cell, B cell, macrophages, dendritic cells. Small energy, the energy. Gives you energy. Delta G. Delta G is equal to delta H minus 2 times delta S. Energy available to the world. Great memory, strong memory with the B memory cells. Receptors and self-recognitioners. Receptors and ion channels. Nerves. Reverse transcriptase. Positively charged. EA sports. Transition state. Towards a side with less moles of gas. Relaxed state. More affinity. Relaxed state bound to oxygen. Keck, the equilibrium constant. I just stuck the con at Keck. I don't really care. Host cells. T cells still. Oh, but these are cytotoxic T cells. Oh, I see. 1 times 8 is 8. Cytotoxic. 2 times 4 is 8. T helper. I think that's what it's trying to get at. So towards the heat, towards the heat. Towards the heat, towards the exothermic reaction. Translation. Exercise. Side chain interactions. Cooperativity. No change in the direction of the reaction, because this is kinetics, not equilibrium based. But all this is equilibrium based. So CD8 times 1 is 1 MHC1 cytotoxic T cells. Oxidative phosphorylation. Kinetic parameter. Increase the temperature, we need to shift the direction towards endothermic to then go more cold. No shift in concentration, because that's mentioned, which is kinetics, and not Le Chatelier's principles, which is equilibrium based. Le Chatelier's, I'm probably saying that wrong, but I don't care. At this point, there's like a lot to remember. Like all the little minor details, who cares? Disulfide bonds, and heat can disrupt these bonds. We got water, and water is used and lost. A shift to the right. Cysteine residues. Alkyl factor, such as hemoglobin heme, which used to bind, contains the iron which is used to bind with CO2. Hydrolysis or dehydration reaction. Delta G prime is Gibbs-Free-Monarch equation, which states that at some point in time in the reaction, under standard conditions, such as 25 degrees C, 1 atmospheric pressure unit, which is 760 millimeters of mercury. 1 molar of all species, including products. Yeah. Oh, yeah. Changing gives free energy. I think it's fine. Positive cooperativity. Oh. Oh, never mind. Nucleus. A left shift. Transition state. Lowering the EA. I don't know what the heck you're talking about. The signal sequence, sometimes called a localization sequence. Nuclear localization sequence. Telling a protein to go to the nucleus. Okay. Sometimes use the term nuclear localization sequence. Again! Again! Substrate concentration. High Km, low affinity. High Km, low affinity. Low Km, high affinity. Negative. CD4 times 2 is 8 MHC2 T helper cells. Positive. EA Sports. It's in the game. EA Sports. It's in the game. I don't know why I think of it like that, but I do. EA Sports. It's in the game. Okay. Exons. Why, dude? Just let that go, man. The signal localization sequence, or nuclear. Oh, I guess just a signal localization sequence, or signal sequence. Now we'll go again. Wobble, 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 wobble. Timing. The adaptive. In the ribosome or in the RER? I guess ribosome. Well, ribosome. I keep saying that. It's just ribosome. Primary active transport. Move molecules against this concentration gradient. Lysine. Why are you so lysine, man? K. Lys. LYS. Lysine, oh, lysine. Nerves. Nerves. Get on my nerves, man. Signal sequence. T helper marked by CD4 plus. Isoelectric point. It's fine, dude. Threshold potential is reached. Yeah. Crank out a ton of those antibodies, man. Turn into a plasma cell. The energy available to do work. Delta G is equal to delta H minus T times delta S. Kind of easy for now. Increase the pressure of the system, then we have to shift directions of the side with less moles of gas. Reason for that is as pressure shifts and as pressure increases, volume decreases, and shifting towards the centrifugal gas allows for equilibrium to offset the new insult. Did you just insult me? Yes, I did. What are you gonna do about it? We're gonna go with the side of less moles of gas. Towards the side with less moles of gas. How dare you? Cytotoxic T cells marked by CD8 plus, CD8 plus. Kill cancer cells and viruses. Yeah, please add that plus in there, man. What are you doing? What are you doing? Organized by MHC1. Cadet face right, right face, right face. Anyways, increase CO2. No change, no shift, nothing happens. No change, no shift, nothing happens, buddy. Sorry to tell you. Sorry to tell you. D. What the heck is D? Oh, 2,3-DPG. So that's how they're trying to trick you, huh? Neutralize free radicals. Create intermediary byproducts, such as H2O2, radicalized hydroxyl, and radicalized O2, I think. Proxyls break down fatty acids, amino acids, and detoxify chemicals. Proxyls use redox enzymes to neutralize free radicals, which help break down fatty acids, amino acids, and detoxify chemicals. Reason for shift is that's McHale's-Method kinetics. McHale's-Method kinetics is about increasing the rate, the reaction rate, but not having some sort of change in equilibrium, which is Lechateliers. Response in equilibrium. Changes to equilibrium. Response to change in equilibrium. Response to change in equilibrium. To a specific organelle structure. I don't know why I call it organelle structure, but that's fine. CD8 plus 8 times 1, MH1, cytotoxic T cell. Glycine. Hello, glycine. Hi, glycine. Hello, glycine. Cooperative. Relaxed. Relax. Take some O2. Intermembrane, mitochondrial membrane space, intermembrane space. CD4 times 2, MHC2. Taut states uptight, not bound to O2, low affinity for O2. Linear sequence of amino acid sequence, linear chain of amino acids. Due to backbone interactions. Helical and beta pleated sheets. MH1 cell. An allele, a mutation, an allele. Secondary structure. Negative cooperativity. CD4 plus. Signal sequence. CD8 plus. Organelle structure. MHC2. MHC1. The forebrain becomes the cerebral cortex. Cerebrum. The stressors can trigger the body's endocrine system and fight or flight, which is a sympathetic nervous system. You flat pig. Follicle stimulating hormone, luteinizing stimulating hormone, adrenal cortical, adrenal cortical. No, I can't believe you've done this. Adrenal corticotropic. Corticotropic. Why do I always miss the last part? Adrenocorticotropic hormone. Thyroid gland T334 hormones. Thyroid hormone T334. Pig, prolactin, endorphins, and growth hormone. And then, oh wait, what the heck? Oh, I'm not even reading the question. Posterior pituitary hormones, ADH and oxytocin. Yup. Posterior right here. There's the only two. Posterior right here, there's only two, and then this one. Dang. Excuse me. Cerebral cortex and synapse on the lower motor neurons in the in the stem. In the brainstem or the spinal cord? Let's go. Okay. ADH. ADH and oxytocin. ADH are those vasopressin. Jeez, man. Jeez. Okay. What's the game plan for tomorrow? Let's figure this out. We could figure it out tomorrow too, I guess, but I just want to get straight into it. Oh, that's so good. What do we do tomorrow, man? What's the play? What's the play, guys?
Keeping the lights on.
No, I won't play no ass. Get out of here. I think we should do that. Okay, let's do let's just do his videos first, eh? High yield sciences and applying the high yield sciences. Wow, that's a long video. Applying the high yield sciences.
You buy Kotex balance pads have a
There's a lot of people that, because the MCAT's composed of scientific articles, it's really different.
Yeah, that's what I'm doing tomorrow. Okay. What else do we do? Oh, that's right. He has to do the actual stuff, eh? EV2, PS2, CP3, cars from Jack Weston. And some of these videos I have here. What? This is an ad or is this video? No, it's a video. I think all this stuff is just like, no.
So this will be the second.
Hold on, buddy. Yeah, some of this stuff, eh? So I wanted to get a better understanding of it. Like I said, the basics of everything. Maybe we can do a little biology archive. up until a certain point, right? I mean, I'm not gonna do everything, and that's just insane. That's just insane. But definitely see what kind of videos there are there, you know, to see, just to play around with. What is this one? Respiratory health and medicine, huh? Hemostasis. I definitely want to do that one though. What is nerve regulation? Not so much stuff though. Maybe a little bit pressure stuff. I don't want to go too much into all this stuff really. Too much medicine that we've you know, that I'll cover at another point. It's so nice though. I do want to go over all this stuff. This is fantastic. And this is great. This is amazing. Amazing. The amazing mazo. Oh, too much clicking. Okay. What is this? The brain? Yeah, I'm gonna watch it again. But I thought I had the brain somewhere here now. Where do I have the brain? Did I get it out? Maybe. I don't know. Anyways, we'll leave that there. And then this is health and medicine again. So we can get rid of this. Health and medicine yet again. So I think did I watch this? Did I not watch this? Oh man, maybe did. I guess we'll have to watch this again. Electronic. This is the ones. What is this? It's the same thing as here. And then this is oh, especially this one right here. What is this? And we'll go through this as well. I think we can do it. I think we can do it. Not too. I think we can do it. And yeah. Yeah, and I guess they're not because it's duplicated. And I'm gonna go over this again with him. So, and then let's just get rid of this. I don't know. I mean, I have it. This is not time to be distracted. Okay, so we have immune system. I do want to go over with him, and let's see, biochemistry of hemoglobin and myoglobin. Biochemistry aspect of it, yes. Let's see what else he has. Kinematics. Oh, he has kinematics. Wait, kinematics? Oh no, kinematics. That's not. No. And this one, I wanted to go over genetics. Genetics. Oh man, do we really? Yes. Yes. All right, Andrew. You win, buddy. Whoa. 290 at 10 inches. How much? Yeah. Wait, what? I think I did the math wrong. Four days. That might not be that bad. I don't know. DNA, I can do like way there. Let's try this one. No, I haven't. Maybe some of the stuff can be cleaned up tomorrow after I submitted the applications. All right, guys. Yeah. There might be. That might be it for now. Let's see how it goes. All right. Thank you for watching, everyone. Have a great rest of your day. Have a good night. Hope things going well. Hope you're studying good. Good luck on everything. Take care. See you guys tomorrow.